microalgae, overcoming the technology's barrier. Bearing the unique qualities and enormous potential, the microalgae have been an attractive candidate for the bioresource material, for the nutraceutical, pharmaceutical, and biofuel industries across the globe, and believed to provide a green alternative besides the environmental sustainability. Many valuable metabolites present in microalgal species remain still unexplored, and this does not only result into economically unfeasible process, but also affects the transit of microalgae from research to viable commodity for biofuel and other bioproduct drawn under a biorefinery approach. The major hurdle for microalgae utilization for various applications is the low efficiency along the value chain from harvesting, dewatering, extraction, and purification, apart from the development of effective and economically interesting microalgae cultivation systems. A deeper understanding of the aspects related to microalgae cultivation is hence needed so that they can be promoted and exploited in a better manner. To address these issues, TERI Deakin Nanobiotechnology Center, known as TDNBC, through DBT TDNBC Deakin Research Network across continents for learning and innovation, known as DTD RNA Network, has organized this workshop. Within the Government of India, Department of Biotechnology, which is within the Ministry of Science and Technology, has been a pioneer, leader, and path breaker in translational research across India. TERI Deakin Nanobiotechnology is standing at the frontiers of nanobiotechnology, which allow it to be a lot more effective in agriculture and environmental sectors. Deakin University, Australia, are leaders in material sciences, and these three institutes, DBT, TDNBC, and Deakin, have jointly created DTD RNA Network. The network is creating joint labs as country hubs and network of research institutes across all continents to develop International Center for Translational Research for research training and education in nanotechnology. DTD RNA Network has many institutes and industries already on board globally with integrating expertise. It includes International Iberian Nanotechnology Laboratory based at Portugal, and this workshop is being jointly organized along with INL. I greatly acknowledge the support of Department of Biotechnology, Government of India for funding DTD RNA Network project under which we are organizing this event. DTD Network is a platform where we are connecting all the nanobiotech community, including researchers, industries, startups, investors, funding agencies, students, and other academicians. All of you are invited to join this network, and do mail us if you want to join this journey. The workshop aims to contribute to improve the economic viability of algae value chain via research focusing on extracting multiple products from algae. Leading academic researchers and technocrats from industries are participating in our workshop, who would share in this e-workshop their knowledge and experiences on various aspects related to microalgae cultivation and downstreaming, which will be supported by lectures and demonstration-based video clips. I believe this is a first event of this kind where the wet lab protocols are being shared online in order for us to continue enriching our knowledge exchanges and the exchanges of views, and we are able to discuss science in a more lively manner. I express my gratitude to all the speakers for taking out time to share their knowledge with all of us. I sincerely hope that the discussion in this workshop will empower the researchers, innovators, and regulators in pharmaceuticals, bioremediation, agriculture, and food industry to achieve a greater understanding about the microalgae using high-end technologies. I welcome you all for this event, and during next 2 days, We would be looking forward for your valuable contribution. And we are definitely excited having seen an enormous enthusiasm among the participation. They have been writing to us, they have been connecting with us, and we have in total 450 registered participants for this event, which is definitely overwhelming. I will now request my two colleagues, Dr. Amrit Preet Kaur Minhas and Dr. Ruchi Agrawal, to take on the sessions and guide you through with some advice, do's and don'ts, so that this event remains very useful for you and you are able to actively participate. We would like to provide you opportunity for greater interaction and discussion, and that's how we have catered this event for you. Thank you and welcome once again. Over to you, Amrit.
Yeah, thank you very much, sir, for the wonderful welcome remarks. So now moving further, since we have participants from various regions of the world amongst us, so we would like to share some instructions that need to be followed for smooth running of the workshop. Before we start our first session, I will now read out the instructions to our participants. We have disabled the video streaming and audio rights for all, except the speakers, to minimize network congestion and maintain the speed and flow of the talk. If you want to speak, I request you to please use the hand raise option. You can find this option in the chat window against your name. Once you raise your hand, we would be able to give the audio right to you and ask you to speak. While you are welcome to raise your hands at any time, we would appreciate if you could please keep noting down your queries and ask them at the end of the talk or during the discussion session. This is to ensure uninterrupted flow of the presentation. We understand that we have more than 400 participants in this workshop. If we are unable to take all questions, we request you to send your questions via email and we will get back to you with the answers. Participants who are joining through YouTube link may also write their questions in the comment section and we will revert with the answers. Now moving towards the session one, which is new developments in metabolites processing, algal biofuels and other commodities, where experts in the field of algal biology will share their recent accomplishments with participants to further the society's understanding of the existing state of the art on algal biology, biochemistry, omics databases, and other resources to identify the challenges. So now, in this session, our first speaker is Dr. Pawan Jatur. Dr. Pawan Jatur is currently a group leader, omics of algae group at ICGEB New Delhi. He had obtained his MPhil PhD from Pondicherry University, India, in the area of biochemistry and molecular biology. Later continued his research as postdoctoral research associate at University of Nebraska, USA till 2012. Joining ICGEB in 2012, his primary focus of research is understanding the dynamics of microalgal systems through integrative multi-omics approach in coherence with reconstructed genome-scale metabolic models with well-defined functional pathways that will elucidate an effective strategy for converting light carbon source to biomass, biofuels, and biorenewables, B3, for sustainable futuristic solutions. So now, I'll request Dr. Pawan Jatur to start his presentation and share his thoughts on this area.
Thank you very much, and I would like to share the slides from my computer. So can you give me the share access, please?
Yeah, please.
Okay, hope all the hope you can see the slides.
Yes, these are visible.
Okay, thank you.
Yeah.
Okay, so yeah. Okay, thank you very much. Very good morning to all the participants, and I would first thank the organizers for designing a beautiful program and other commodities. So I would once again thank the organizers for inviting me to share our research, what we are doing at ICGEB. So as part of introduction, I am Pawan Jutur, group leader at Omics of Algae Group, and so this slide is the one. So what we can do with the microalgae with minimal information. So those who are interested to do microalgal research from the beginning, they have a huge possibilities of understanding the microalgae. So I am trying to cover most of the things. Mainly, we'll talk about biomass, biofuels, and byproducts, how microalgae can be characterized on a omics perspective. And on the background, you can see the first designed microchloropsis Gadiatana rubisco, which has been designed in our lab. So to go ahead with that, I will just briefly go what is ICGEB and what we do at ICGEB. So ICGEB is a intergovernmental organization which works on field of life sciences and biotechnology, where we have three different components, especially our headquarters is located in Italy, and presently I am present in India, and one more campus we have at Cape Town, South Africa. So we have broader areas of research where we do infectious disease, non-communicable disease, medical biotechnology, and I work in the field of industrial biotechnology, where we are trying to understand the biofuels and their relevance in context to general biology and also a transformational research. Other program is plant biology and biotechnology, where and we have different member countries who participate in research and other activities. And ICGEB also involved in meetings and courses and also funding for people around the world, especially for member states. So this is a glance what we do at ICGEB. And going to the talk, I would like to have a brief introduction because we have a lot of students in this webinar, so workshop. I would like to start with biofuel generation. So everybody knows that biofuel is a catching up area in terms of not only a solution for a future, but also a sustainability factor is required to get clean energy, clean fuels in the near future. So in order to have what will be the constraint of biofuels and what kind of generations we are talking about, this is a very good representation of how the biofuel generations are represented. And first coming to, there are 4 generations of biofuels. The first generation is especially edible crops, which consists of sugars, rice, wheat, potato. And the first generation biofuel is not a sustainable because we cannot compete or we cannot be a competition for the food resource. Food is equally important resource. The second generation biofuel is the one which is more interesting and has a great attention, and a lot of people are working on that, where they work on waste lignocellulosic biomass, especially forest residues, sugarcane bagasse, cell biomass for fermentation, all this. And the third generation is especially the microalgae, which is the biodiesel precursors for other things. And fourth generation is how to sequester the CO2 in order to produce clean biofuels using CO2, jatropha, castor, and all non-edible source. So these together makes a raw material selection, which are very important starting material for a biofuel generation. Then followed, once you have a raw material selection, you can select the raw material, go to the second part of that, where you have to convert. So technology or transformation is the main importance, where you can convert these raw materials to the more relevant chemicals or biofuels what you are expecting. So mainly the technology transfer is through maybe physical, it may be through chemical, thermochemical, or biochemical. So these technologies may be discussed in later eminent speakers in this workshop. So I'll not be talking about that. And finally, the product selection. So we have a range of products which can be generated employing these raw materials that are bioenergy, biofuels, biomaterials, or biochemicals and food products. Thus, you can have a multiple product range using these biofuel generations, and the sustainability is the one challenge which we are looking for, and that is where it is very important to understand the biology of the biology of any system what we are working upon. So in a context, just for need, why do we need energy resource are ever increasing CO2 released from the fossil fuels causes climate change. We need clean fuels as it is a major concern, and lipid petroleum transport fuels especially cannot be replaced by renewable sources like wind, solar, and hydro. The main point is all these biofuels are carbon neutral. So the net energy is either negative or zero. That is making us what we call as a clean energy. So again, in that context, because our work concentrates on third generation biofuels, I'm going to discuss more on algae. Why algae? So third generation, as I discussed, the third generation biofuel, algal biomass derived a great promise or great advantage like high amount of oil productivity per acre. They can be used in non-arable land. So there was a questions from the participants that were because India is having a huge coastal belt, and that can be used as a non-arable land with marine waters, and variety of water resources can be used. That's advantage, and also it mitigates the green gas emissions, which is a very primary important for environmental constraints. Beyond that, the good potential for using these is they not only produce biofuels as a major product, that they also produce a lot of valuable co-products or commodities, which are very relevant or useful products of what in a pharmaceutical, nutraceutical, or any kind of industry. So what are the challenges in a nutshell? So we, as Dr. Alok was mentioning previously in the talk, there are a lot of challenges which has to be, the whole technology of biorefinery should be economical and also sustainable. So in order to do that, these are the major issues where your large scale production of how you can produce the algae, where the nutrient supply, gas exchange, photosynthetic activity, all these parameters have to be worked upon. Along with that, harvesting is the cost to the microalgal refinery. So putting all these together is leading to the high cost what is at present, where we are not competitive enough for the fossil fuel. So if we can work out in these directions, where we can engineer different methods of harvesting, large scale production, that definitely will bring down the cost of these things. So in order to illustrate, always I try to emphasize like everybody has a question in mind, is algal biofuel sustainable or not? So this is the best example where a consortium from NAB report, which was recently published in 2016, is a group effect showing that there is a road to produce the microalgal biofuel at gallon at the rate of $7.5. So this is a beautiful roadmap which explains you how you can start up your work and do different kinds of things. Important thing is, if you can see here, this is a discovery where a lot of labs are working on that to look which is the efficient strain, how you can define the efficient strain, and those can be cultivated in either open ponds, closed ponds, or different types. So the major thing where they have shown is the harvesting, and they used electrocoagulation, and they could recover 95% recovery efficiency. So along with that, they are working on improving this so that the cost can go down. So without adding the cost of co-products such as fish feed, nutraceuticals, all these things, they have calculated that they can produce the biofuels at a rate of at the gallon from $7.50. So this can be still reduced when you add the cost of the other commodities such as high value products. Definitely the cost of biofuels can be competitive enough and can be sustainable. So this is a kind of a nutshell, and it is available online. And this is an interesting review or article which tells you the best way of analyzing where you are and what are the steps you can do to improve the each step so that you can produce sustainable biofuels and co-products for the futuristic world. So we call, so at ICGEB, we work on all the aspects of microalgae to understand the general biology. And so kind of we develop a pilot scale model to take it up for the industry so that that can be scaled up. So we call the thematic as B3 algae, which is biofuels, biomass, and biorenewables. So different colleagues of mine work in this area. So especially area where they're interested to enhance the biomass using different microalgae with different conditions. So other students work on omega-3 fatty acids, biofuels like biodiesel precursors and other things where they are interested to enhance the biofuels. And third group where they work on bioproducts, where they work on different types of bioproducts, where squalene, they work on different carotenoids like lutein, astaxanthin, beta carotene to enhance those. And we have a team of bioinformatics where they work because we generate a lot of omics data. We try to interconnect those data to understand so that the biologists can make a better conclusion of what to enhance, what not to enhance, so that metabolic or genetic engineering of a particular strain can be done to enhance those things. So we call it as integrated omics concept or iomics concept. So this is a nutshell. The omics revolution is a integrated comprehensive omics approaching combining the genomics, transcriptomics, proteomics, and metabolomics for understanding the molecular mechanism of the cells. So the main, if you see onto my left panel, you see that there are usually the traditional way is we focus on one molecule or one pathway to engineer those things, but really on a constraint. So when we see a microalgae in a natural environment or at a laboratory scale or a high scale open pond reactors or closed pond reactors, this is not what is happening. So we can produce, as I said, not only producing biofuels, we can see for other commodities. And so the disadvantage of single omics is at each omics level. For example, when a particular transcript upregulate, it is not mandatory that the transcript can be translated into that protein of interest and corresponding to a metabolite. So it gives a very vague experience that if the particular transcripts are upregulating, it does not mean that the protein also should increase. So it is very important to have an integrated or multi-omic approach where each omic layer crosstalks with each other in order to give a better outcome of what is happening from genomic to metabolomics. So there was again a question from the audience like whether we can do just this thing. You can do without genome background, can you do any kind of omics to understand that? Yes, this is the solution. So we have two models, what we call as forward omics and reverse omics approach, which is called feed forward approach or feed backward approach, where if you have the genomics information like classical forward genetics approach, if you have a genomics information, you can do the transcriptomics, correlate and cross proteomics, and then your metabolomics altogether gives you forward genomics approach, omics approach, where you can understand the system. So in that way, in that context, like for example, for chlamydomonas, you know the genome of chlamydomonas. So in that way, you can just start with any of the transcriptomic or proteomic, and you can integrate with your genomics, making it iomics approach to better understand what is happening with a particular condition. Whereas in reverse omics, it is entirely different. So genomics and transcriptomics with proteomics is still an expensive area where you have to invest a lot of time, money, and also expertise in that area to integrate the data so that you can generate those things. But for the only solution here is you don't have a genomics information. You can take the metabolites, you can extract the metabolites, you can quantify those metabolites and map on such a way that I'll be showing one of the example what we do in the lab, where we from the reverse omics approach, how do we go for a solution further. So that's if you know the metabolomics, you can map further and go towards proteomics and transcriptomics, integrate towards that and understand the whole thing. So it is very important with the tools available nowadays. It is very possible to not only have all the information, but without no information, you can starting up, especially who are interested to start from the scratch or who want to look for just how the different changes happens within the environmental constraint, it is very important for them. So these are the few publications which have come up recently in our group, and these are available online. If anybody wants, I can definitely share with them. But this tells the first approach where we are talking about biomass, how to enhance the biomass. So the rationale here is we are understanding the cellular dynamics of microalgal cell factories subjected with carbon supplementation. So it is important that you can enhance the biomass, how you can enhance the biomass, and understand through multi-omic carbon conservation mechanism or the activity of photosynthesis towards enhancing the cell biomass. So this is what we have done. So this is a recently publication which has came up in Frontiers in Plant Science, where we are trying to understand the carbon concentrating mechanism is Microalgae gadiantina when it is subjected to very low CO2 and very high CO2. So we know that CCM is very active at very low CO2, but because these marine species are located at very low CO2 concentrations, and they have developed a mechanism where they don't waste any carbon which is available in the near place. So we are trying to understand through omics approach, and you can see in my left panel where you can see in a high, the yellow line or yellow is indicated by high carbon concentration, which is around 30,000 ppm, and this is around 300 ppm, which is very low carbon concentration, 0.03%, which is present in air. So we want to have, and we want to have how the biomass gets enhanced, and we want to have a solution for that. So this shows how your biomass doubles, and the different chlorophyll and carotenoid contents increase in course of time when it is carbon is getting diverted towards different molecules. So, and also when in VLC conditions, what is happening? It is very important to understand at biology level how the organism is behaving with carbon, with nitrogen, and carbon and nitrogen ratio plays a major role in these things. So to understand that, we went for feed backward omics approach, where we took the metabolomic samples of the species, and we try to separate these. We try to precipitate the proteins out and take all the metabolites, derivatize those metabolites, run the metabolites by GCMS or LCMS approach, and then finally identify these metabolites through the library, niche library, which is available, and then map onto the software where we can quantify these all the automated data analysis or data analysis can be done. So this is how once we run these conditions, and we get this heat map where they consist of different fatty acids, sugars, alcohols, amino acids, organic acids, and other compounds which are important, and we can also see which kind of metabolite is upregulating and downregulating with respect to drugs. Thus, in a nutshell, so we have tried to understand not only what is happening at carbon concentration mechanism, how it is enhancing, where if the carbon is getting through Rubisco cycle and it is enhancing, especially the sugars like maltose and fructose, and also it enhances trilose, which is captured, and whereas in high carbon concentration, the Rubisco is fixed in such a way that it enhances not only the fatty acids and other things, it also enhances high value products like alpha tocopherol through terminal responses. Thus, we can understand, and we have overlaid, and then we went to a proteomic approach where we have done a proteomic analysis. It's called qualitative proteomics, where we have done the whole cell proteomics. Then when we have seen these proteomics, so there are different things, and especially to capture different proteins, we try to, because we know the CCM mechanism is very active within first 24 hours. Initially, the experiments were done for 0, 3, 6, 9 days, because to understand how the metabolite regulation is happening. Once we have a conclusion that you can select the particular time points in order to evaluate, because what is happening at the initial time is more important, because we are understanding how the biomass is upregulating or downregulating. So in order to understand the CCM, it is very important to capture 0 to 24-hour cycle, and this is the differential expression of different proteins involved in VLC and HC conditions. So thus, in the context of that, this work is being carried out, and because I'm not discussing much of details. If anybody has questions, I can do or definitely answer. So this is what the final conclusion of schematic representation, what is happening between first 24 hours. And we can say that the green part is the one which is indicates that it is getting upregulated during the green arrows or a green color, which indicates they're upregulating during VLC, and the blue indicates they're upregulating during HC conditions. Thus, yes, as I said that in VLC condition, the pentose phosphate pathway goes up, photorespiration goes up, the CCM genes goes up, and it gets fixed up into sugar. Same wise, the TS cycle enhances, and it within next. So the gray scale is 16:8, which is a very convenient period how you have to do a time course. And this dark region, this is a dark period, and the light period is for 16 hours. And we can see that different high value products along with the lipids are being generated. So now we have different targets which have been established, and when we go crosstalk with our metabolome data, what is happening with the metabolome upregulation is also being reflected in proteome. So this is the first time what we have done is we have reconstructed the structure of Rubisco of Microchloropsis gadiatana using homology modeling, and this work is still going on. The intention of this work is we want to understand how the CO2 and O2 competes with each other in order to get fixed with their active sites when there is a competition between them, and how they can do. So this is where we are understanding through modeling studies what is happening. And this is from our genomic information. So we have taken the sequences and we try to reconstruct the structure of one by homology modeling, and thus it is capable that the omics information is very relevant for us to do different parameters in due course to understand what is happening within the cell. So these are some of the things. So coming to the second part there, where I'm talking about biofuels. This is latest publication. So the rationale here is to understand the nutritional stress, and this is a very popular area. Everybody in microalgae research knows that, because when you remove the nutrition from the algal species, it can accumulate a lot of neutral lipids, and that can be increased. But we are compromising with the biomass. So is there any way to understand the biology, how it can enhance the lipids, but not only without compromising the biomass, is our basic agenda. But here I'm going to discuss what is happening with the nutrition stress, why oil is getting accumulated in context to a multi-omics approach, where we try to do end-to-end feed backward omics approach, starting from metabolomics and integrating with genomics to give a complete picture of iomics and understand what is happening with the targets of lipid biosynthesis. So in a nutshell, we have selected a marine microalgae, what is known as Parachlorella casleri. This is an indigenous strain, and we have subjected that strain to deplete conditions where we have seen that it can enhance the lipid biosynthesis, but the biomass is compromised simultaneously. The photosynthesis and beta oxidation gets also compromised. So when you see these cells, basically this photograph is of chlamydomonas. You can see that a lot of lipids get accumulated in these contexts. So when you replete back these cells, they can become normalized, and lipid biosynthesis, that is neutral lipid biosynthesis, goes down, and photosynthesis and beta oxidation comes. This is very important in order to sustain, because when the lipids are getting formed and you can extract or milk out these lipids, can you retrieve back the algal cells so that your seed culture is normal, it becomes normal, and you can use that culture again and again to produce multiple. So it is a very good area of to be explored, so that you will have a sustainable growth profile of microalgae at any context. So what we have done is we have done a stress biology where this data is available in biotechnology for biofuels. So what we have done is we have taken the Parachlorella casleri, as I said, and subjected to different, we try to remove each of the nutrients, that is nitrogen, phosphorus, and sulfur. And you can see in my right panel that the lipids get accumulated when we remove the respective nutrients. And this is the lipid profile. And we have seen that the lipid profile in nitrogen is very higher, and there is even there is a compromise in biomass. But whereas in phosphorus condition, even the lipid is very high, but the biomass is not compromised. That's why there is overall productivity goes down. So that is not a problem, and this shows that this is a different composition of pupa, mupa, and safa, where it is very important to understand that biodiesel components require less of pupa and more of other components. So it is very important to analyze what kind of profile is there. But nowadays there is a method where hydroliquefaction, all these methods, if you can do that, directly the crude oil comes out, and that can be fractionated in different forms. So it is very important what kind of fatty acid profile is present. So so what we have done is, as I said, like we went for fed omics, feedback omic approach, where we have done qualitative omics. And as I said, as explained in the previous slide, we could find out like multiple metabolites which were upregulated and downregulated during the course of time, and these were evaluated. And we can see that this is hypothesized from that the during N, the red means that nitrogen, and blue means phosphorus, and S is yellow, where the arrow indicates they're upregulated and downregulated. You can see the multiple metabolites get upregulated and downregulated, and the red is indicated by upregulation. You can see that the pyruvate and the NADPH, which is increased to fatty acid biosynthesis, along with acetyl-CoA ACKs. So along with this, there is an increase in glycoprotein metabolism also, where this is contributing. All the other pathways are downregulating. Somehow, with the metabolome approach, you can also look forward what is happening to the your metabolic profile. Thus, you can understand how the overall process happens when it is subject to particular condition. This is very important, and a time course experiment is very important to understand that. So afterwards, we went for proteomic approach and try to understand how many proteins are upregulated and downregulated. We found that there were nearly 2,000 proteins, and common proteins which were upregulated or downregulated were 483, and significantly 166 proteins. So we are talking about the 166 proteins, all the proteins which are not only lipids, but we have only 10 to 15 proteins which are upregulated, and 100 transcripts which are nearly upregulated and downregulated when it is nitrogen is removed. So in order to understand the transcriptome, we have done Illumina sequencing, and thus we have found that when we overlay all the transcriptome data on the KEGG mapper, you can see the red and blue, where the genomic and transcriptome is the blue one, which is integrated together, and the pink one is the one which has other metabolite pathways. So we have integrated the genomics and transcriptomics in order to understand. The insert here shows different kinds of transcripts which are upregulated and downregulated. We have evaluated whether what is happening with the transcriptome data has been reflected, and so we can see that what is happening with our transcriptome data is also being evaluated through qPCR experiments, where the fold change can be shown here. So these are the targets where they are upregulating. So if you try to overexpress this kind of transcripts, the proteins also get upregulated, and you can increase the biofuels. And these are the ones which are downregulated, and so these also can be worked out what is happening when you can knock out. So we have done the knockout of these particular transcription factors, and we have found that there is a compromise in biofuel. Thus, we can confirm that based on the multi-omic data, we have identified what are the different transcripts or proteins which are upregulated, and based on protein and transcriptome data, we have integrated a summarized how the central metabolism and nitrogen deprivation leads to the production of fatty acids, that is oil, and finally, and the red here shows the transcripts which are upregulated, and blue shares. So interestingly, you can see that the TCA cycle goes half. That is, so we call it as half TCA, where the complete TCA cycle is not being observed, and downregulation of all the things goes down, and it bypasses through fumarate and malic and to pyruvate, so that pyruvate goes to combines with acetyl-CoA and forms lipids. So thus, this is what it happens in all the microalgae respective. But if we have to really evaluate what are the transcripts and proteins which are upregulating and downregulating. So based on that, we have done the whole genomic approach, and this paper is out recently in November 2020, where we have done first time the hybrid genome assembly of Parachlorella crocellari, where we have done nanopore and Illumina to integrate the data, and you can see this pink pathway is the lipid biosynthetic pathway, and the green all the pathways which are corresponding are very much conserved, and we have identified the pathways which are present in that. Integrating all the data is really a messy visualization where you can find number of transcripts, genomes which are upregulated and downregulated. Thus, you can understand if you are, want to increase the pathway, what are the things you have to do, what are the things you have downregulated, and the yellow dots, what should represent the metabolites which are upregulated and downregulated. Even thus, you can integrate from genomics to the, and this is respective to nitrogen starvation when it is subjected and how the lipids are formed, and thus you can do a time course experiment for any kind of this thing to understand the biology of that, what is happening. And that is what, if you can understand, you can really media engineer the system or a genetic engineering system so that you can produce the interest byproduct what you want. So this is the third area what I want to discuss, which is nothing but byproducts, and these are the papers where we work on tocopherol and other mixotropic this thing. So here, as I told you, that we need some carbon substrates to enhance the. So for an example, today I'm going to talk about omega production, so which is a very important nutraceutical product, EPA and DHA. So how we are enhancing the thing using the varying carbon substrates that we are going to discuss today, and how we are going to enhance that. So the other byproducts such as lutein, so I can discuss. If anybody is interested, they are welcome to collaborate and interact with us. But for example, how omics study can give us a better rationale for how to evaluate these pathways for production of omegas, I'm discussing today. So this is a global market, and we know that by 2025, the global expectation is around $3,451 million for omega-3 fatty acids, and different companies which are already producing. And we have one Indian company also in this where they are able to produce omega-3 fatty acids. So these are the very Paris India Limited, which is located in Chennai, and there are a few companies which are already in this market. So it is very important to understand how to enhance omega-3. Why omega-3? So algal omega-3 has a huge attention, and these are different byproducts which can be used. And not only that, very interested like the DHA, which is being supplemented in Bonvita. Little chance is nothing but a DHA-rich algal oil. So it is isolated from algal oil. So it is very interesting that we are already have a huge market space from microalgae, how it can be, but there are some things which has to be worked out. So thus, these are different kinds of products which are already available, and people are already using those things. So in order to understand that, what we have done is our rationale here is we have an indigenous Oranchochiton species. It's a heterotrophic algae. So where it can use the sugar molecules, carbon as a substrate, and it can produce biomass and DHA production. So this is an oleaginous microalgae, and so basically we cannot get glucose at an industrial scale. So we were looking for different substrates, simple sugars like glycerol, xylose sugar, and all. And we have seen that we can use glycerol and we can yield a better growth biomass and DHA productivity when you use glycerol. This table shows that glycerol, because glycerol, crude glycerol is a byproduct of algal biorefinery. So we can get at an economical cost where we can substitute instead of glucose, because glucose will not be cost effective. So we can take the crude glycerol as a substrate, and we can subject that for enhancing the omega-3 fatty acids. So in order to do that, so this is a microscopic representation how when you subject to glucose or glycerol, there are large bodies of lipids which can be seen within the cells, and this is a body piece training where you can do fluorescence dye method to understand what is happening within the cell. So you can see in the glycerol there are huge lipid bodies which are being formed. In other things also you have lipids, but very low. It's at low concentrations, but the biomass is get compromised. So we cannot compromise on the biomass when we want something along with the byproducts. So what we have done is we have done some metabolomics approach where we try to understand, and you can see, as I said, like number of metabolites get upregulated and downregulated. We have found number of sugars, fatty acids, organic acids, which were found in different carbon substrates, and we selected glucose and glycerol for further to understand what is happening with that. So in this context, we can summarize that the overall DHA gets enhanced, and these arrows indicates that different metabolites get upregulated during this pathway, and overall the DHA productivity increases without compromising the biomass or even increasing the biomass. So then what we have done is we have used to illustrate what kind of pathways are getting, and roughly around 2000 proteins were found in that protein profile, and 800 proteins were downregulated, and 350 proteins were upregulated overall. And you can see that the red indicates the upregulation, blue indicates the downregulation, and green indicates no effect. Thus, you can correlate, and the data correlates with that, what is happening there. So basically, in a nutshell, once we do a proteomic approach, what we can do, we have understood that different intermediate of PPP goes up, enhances along with the amino acid profile, where it is contributing to HPL-CoA through malonyl-CoA, and through fast system, the DHA is being produced. So there is recently a paper from a Chinese group where they demonstrated that the use of glycerol and glucose, the PKS pathway activates. There is an alternative pathway called PKS pathway. We could identify the PKS genes, but during glycerol treatment, the DHA production is through fast pathway. So we are looking for a solution where an alternative pathway can also be worked out. So for our assumption that the PKS pathway is working during glucose substitution and FAS during this thing. So we are still evaluating, and oxidative phosphorylation goes up, half TCA cycle activates. So these are the very independent findings and important findings, what happens with that, and how why a particular metabolite or a byproduct of interest is getting upregulated and downregulated. Now we have done the transcriptomics. The study is in analysis part. So once we integrate the proteomics and transcriptomics part, we'll get the targets, what is happening when it is subject to glycerol and glucose concentration, glucose supplementation. Thus, we can integrate, and we can identify how we can enhance the process. So thus, in a nutshell, to finish off my talk, our understanding of biological systems through integrated omics to engineered cell factories. So this is what I have told, that we go for multi-omic approach so that you can understand and come to a conclusion that it may be metabolically engineered or genetically engineered. So that the other work, what we do is we put in a system where we can understand so we reconstruct the whole pathways and try to understand what is happening, as I explained you in previously. And also we do a mathematical modeling, like using systems biology, genome scale modeling, to understand what is happening, whether whatever the data we are generating can be took to system, and we can do fine tuning so that we can give those conditions in order to make the sustainable microalgal cell factory. So this is what we work on, different areas, and I welcome if anybody has any questions or anything, it is in any collaboration interest, they are always welcome. So these are some of the queries by participants where I would like to put emphasis a little bit on. So there are a couple of questions which have been directed, and I would like to just discuss in brief. Has metabolic profiling of microalgae carried out? Yes. So as I done that, as I told you that we work on a lot of metabolic profiling of our microalgae in different ways using feed backward approach, and the other groups also worked on different microalgae which are given here. And very little work is being done, not an extensive work, but there have been some reports, especially from our group in Parachlorella capillary, Microcoropsis, and all those things. The second question is, how is the algal metabolic study done without algal reference genome? This is what I have already answered. So there is an approach called feed backward omics approach. Through metabolomics, you can lay down the metabolic pathways and understand that it is very much possible, and we have developed in our system, and we are happy to collaborate in any means. So the other one is recombinant use of food or feed safety measures. Any recombinant algae which is directly cannot be used for food and feed because of highly relevant regulations. But the extracted metabolites can be used. For example, we are working on one of the products where we want to enhance the astaxanthin from recombinant protein, the recombinant microalgae. But because that product is out of algae, you can use those kind of this thing because it does not have it does not have. So the anything which is directly related for the intake as a food or a feed is right. It cannot be directly taken, but they are certain constraints, and in due course of time, I think there will be regulation. The last question is with enormous. So the reason why we should have indigenous strains which can be cultivated and they can be used in as I said non-edible lines along and with great asset. So there is a great diversity in India for microalgae, but I think this is the right time. If we can recognize, you can understand the biology, it is very easy. Not only genetic engineering is a difficult task in some of the microalgae, but you can media engineering, but that is because these microalgae is already producing different products in natural environment. So we have to fine tune those media engineering conditions so that we can produce the desired products of our interest. So last but not least, I would like to thank ICGEB for constantly supporting us to do this research, and also funding agencies. Without them, it is impossible to do these dream projects of what we can attain for and how you can attain for like DBT, DST, BIRAC Mission Innovation. And finally, I would like to thank all my students, postdocs. Without them, this kind of work is impossible. So with this, I have finished my talk. I'm open for questions.
So before we start the question answer round, so can you share the demo clip which we requested to?
Okay. Okay.
Yeah.
Sure.
After
sure,
that
sure,
we can.
Sure.
Yeah.
Okay. So what I will try to do is I will just explain the protocol for that in a nutshell. Then I will play the demo.
Right. So.
So just bear with me once. I'll just share the slides with you. So can you see the slide, HPLC for carotenoids?
Yes, sir.
Okay, thank you.
Yeah.
So I will just briefly give you the notes how we do this is a very, very important protocol because carotenoid profiling is microalgae is very important, and we are just trying to show you a demo. And if anybody is have any queries, please email me, and we are happy to interact with you guys. And what we basically do is, this is especially for green microalgae. You can take so cell count is a major issue. So you should have at least 10 to the power of 8 cells, which can be harvested by centrifugation. And extraction procedure is you can carry out in 1 ml of methanol with glass beads, where you can do continuous vertexing. And this is also a very important steps where you can do multiple vertexing steps, because some microalgae is very hard to break, some microalgae is very easy to break. So you have to standardize this condition very much, where it is important to do vertexing on multiple sites, multiple times, so that you can extract as much metabolites as possible, carotenoids. Then you can centrifuge for 5 minutes and the supernatant was collected. And likewise, you can repeat the step couple of times. You filter, centrifuge those things and filter through 0.22 microfilters and run through HPLC. So, and should be coupled with UV or PDA detector in order to analyze those things. So you take the, so these are the HPLC run condition. So we are happy to share this because it is not published data, but still these conditions can be standardized at your conditions. And these are the things after multiple conditions. So we use a special column called C30 column because for carotenoids, and the injection volume is 40 microliters with a flow rate of 1 ml per minute. And we do use different solvent systems, and this is the gradient what you use, and the detection rate at 450 nanometers. So once we run the thing, you can see from that this is how the chromatogram looks and what kind of and you run the standards to evaluate what is the particular carotenoid or something. So this is how the peaks come up. Different peaks come up here. Integrate those peaks such that you will recognize. And finally, this is how you can calculate the sample area by standard into standard concentration, into injection volume and dilution. This is important to understand. And these are different kinds of carotenoids which are identified, and you can thus identify what are the kinds of carotenoids. And also that can be the LCMS, where you can identify the new carotenoids which are present in. So based on your condition, you can see that your astaxanthin going to go up, your lutein can go up, all these things. So you have to have this concise how you can evaluate. So this is what Dr. Chetan from my group is going to show you in the demo. So if you have any queries, you're welcome to thing. So now I will share the demo video. So just a minute. So you can see the video.
It is visible. Yeah.
Okay. I hope you can listen to the voice.
No, we are unable to listen you.
No?
No.
Mute. If you find any issue, we can hear from TERI side.
Yeah, please. I think I'm not getting audio here. So you
Good morning. We will be taking sequence to test for 7 to 8 micro cells, and we'll add 1 ml of 100% extraction of carotenoids. Then we will add glass beads for the proper extraction and crushing of microalgae cells. After adding glass beads, we will put this for extraction and the vortexer for 15 minutes dark. After the incubation, we will remove the sample from the vortexer and go for the centrifugation at 10,000 G for 5 minutes at 4 degrees Celsius. After this step, you have to check whether the pellet is totally white or off-white in color. So confirm that it has total pigments has already been extracted into the solvent. If no, then you have to repeat this step and cool the extractants into one tube, and then again go for the centrifugation round. After centrifugation, we will transfer the supernatant to a fresh tube and again go for another round of centrifuge to remove any remaining debris from the sample. After this step of centrifugation, we will filter the extract by using 1 to 2 micron filters and later transfer it to a fresh amber colored HPLC vial with 100% ethanol as a blank. After the sample preparation, we will move to the HPLC analysis. The HPLC run was done by using EZ Chrome Light software, where we will edit the sequence by going to the left pane and selecting the sequence tab. Here, we will edit the sequence according to the plan, and we will save the sequence with respective date. The data storage location will also be set using the properties under this tab, and then we will go for the downloading method and load the samples at the respective location. After downloading the method file, we will go to the sequence tab and start the sequence run. We will wait till we get a response graph. response graph. It takes 35 minutes to complete the run. After run, we will analyze the results by using zip offline and offline mode. And after opening the zip light, we will open the file of the station folder and you will see the results. Here we see the file has already been integrated. So we will put the auto integration off by clicking the tab below and manually integrate the peaks. After manually integrating all the peaks, we will export the data into PDF file by selecting the already made template. Here we will see the results as area percentage, height, and height percentage, the respective retention time. Later, all the peaks will be analyzed based on the retention times, standards, and their respective areas. This is all for the workshop. Thank you for attending.
Now we can quickly take one or two questions from the participants. So we have first question from Dr. Anil Mishra, and his question is, CO2 remains as CO2 or get converted to respective derivative? So this question is to you.
Yeah, thank you. Thank you. And sorry, I think audio was not very clear. We are not professional this thing.
No,
so
it's
I think
fine,
we
sir.
could
I
not.
can repeat.
Yeah. Yeah. Yeah, the question I got it. So basically, the CO2 gets converted into different metabolites, and it is very important. That is what through qualitative or labeling experiments you can track the CO2 when you label it, what kind of metabolites get generated. So basically, the CO2 gets converts into different metabolites. That's what I'm talking. So whatever form of carbon you give, that gets integrated into different metabolites. It may be biofuels, it may be other metabolites. It gets into different products. So CO2 will not be in form of CO2. Yeah. Hello.
How do we predict the exact harvesting time for a particular algae for algal farmer point of view who do not have GCMS analysis approach?
So basically, you can do biochemical assays. So we are also that if you can see our publications, we have done some biochemical, simple biochemical properties, like you can know the lipids, you can know the carbohydrates, chlorophyll, and carotenoid contents using spectrophotometer. So they are the very simple combined assays where you can quantify your different sugars or lipids based on that. So these are well available, and I can share if you want then they are freely available. So that's how you can quantify in a simple way. Then anyhow, if you want to do analytical way, then you have to quantify through GCMS and all. Yeah.
Now, since we have to maintain the timing, so I will request all the participants, if anybody has any kind of questions, so they can share the questions in the question answer section, and I will request that they should not share the questions in the chat box. Instead, they should share the question in the question answers window, and then we can forward the questions to the speaker, especially Dr. Jutur, for this session. And now I would thank you very much, Dr. Jutur, for the knowledge-filled lecture and the information on how to achieve multiple bioproducts and bioactives from microalgae using omics approach. And your comments will be especially helpful to the students who are willing to start their career in this field. Thank you so much.
Thank you. Thank you once again. Thank you for inviting me, and it's my pleasure. Thank you.
So now we move towards our next lecture by Professor Sunil Papi. Professor Papi, PhD in microbiology from Punjab University, Chandigarh, has over 35 years experience in research and teaching, especially on the use of microalgae, cyanobacteria as a biofertilizer and other value-added products. Dr. Papi has developed a commercially viable protocol for BGA biofertilizer production and the technology licensed to a number of industries, leading to its large-scale production and use. Dr. Papi has so far carried out 15 different research projects and has over 85 publications, both national and international, and edited 6 books. And he has been awarded the Best Teacher Award of ICAR-IARA for excellence in teaching and Best Professor in Microbiology by Agriculture Innovation Congress and CMO Global. So now we welcome Professor Papi to deliver his lecture. Dr. Papi, you are on mute.
Okay. Am I audible?
Yes, yes.
Okay, so thank you, Dr. Amrit. I will share my PPT. Are you able to see my PPT?
Yes, we can.
Okay. So at the outset, I would like to thank the organizers, especially Dr. Alok, for giving me this opportunity to be one of the speakers for this e-workshop. And today I shall be talking about some of the work which we have carried out on Phycobili proteins from microalgae or other cyanobacteria. How we really have gone for its purification, characterization, and maybe production enhancement. As you see here that I belong to Center for Conservation and Utilization of Blue-Green Algae, which is located at Indian Agriculture Research Institute, and this is one of the national repositories for freshwater cyanobacteria in India, and this particular repository has been recognized by National Biodiversity Authority. The basic aim of our center is to have cyanobacterial germplasm, especially on freshwater cyanobacteria, and presently we have about 800 unialgal strains with us, comprising unicellular, multicellular, heterotrichous, filamentous, all kind of strains are there. And these isolates, they are constantly characterized for various attributes. And these are also preserved with the best possible means, and our center acts as a repository wherever for research purpose or for any commercial purpose somebody requires some strains, we cater to their needs, and as well as if someone wants to deposit the cultures with us, they will be preserved here. And whenever you require, suppose you are carrying out research on microalgae or cyanobacteria, and you are changing field, or maybe you are leaving a particular place and you want your cultures to be preserved, so this is the best place that you can approach us, and then your cultures will be preserved for long times together. So with this brief introduction, what I would like to talk about, or coming to my topic about biopigments, And these cyanobacteria, these are the major producers of these naturally occurring colorants. And among those, Phycobili proteins are very important. They are further classified as phycocyanin, allophycocyanin, phycoerythrin, or phycoerythrocyanin. And these pigments, they have a very huge market globally, and they are actually, I would say, the cost will depend upon their purity. And these Phycobili proteins of analytical grade purity is around US dollar 13,500 per gram. It's recently it was communicated. And these pigments, these are odorless, non-toxic, and so that's why they are used as natural colorants in, I would say, edible products, food products, value-added foods, beverages, nutraceuticals, pharmaceuticals. And then these pigments are also made insoluble, and they are used in cosmetics and cosmeceuticals. Many of these pigments, since they fluoresce, so they are also used in immunological studies, cell flow cytometry, and many other, I would say, applications. And we have basically reviewed all the uses and application of these microalgae pigments, as well as their enhancement strategies in one of the review we published in Critical Reviews of Food and Nutrition last year. And after pigment extraction, the spent material is used as animal feed or fertilizer or fuel, as Dr. Jutur was talking about the biorefinery approach. These I'm showing here a little structure of these Phycobili proteins and how they are basically biosynthesized. I will not go into the details of these, but just to tell you that these Phycobili proteins, they are open chain tetrapyrrole chromophores, and depending upon their nature, they can be of differently colored. You have blue colored, which we refer to as phycocyanobillin, and of course, because of their color, they have a specific, I would say, absorption maxima. Like for these phycocyanobillin, it is between 600 to 670 nanometers. The red colored ones are phycoerythrobilin, having a maxima between 540 to 575 nanometers. And then you have more of these phycoeurobilin are also there, cryptovalin are also there, depending upon they keep on discovering more and more of these pigments in different kind of cyanobacteria. And the most interesting thing is that the genes, especially the CPCA and CPCB gene, which are basically coding for alpha and beta subunits of these pigments, these are actually one can regulate or modulate these genes with the environmental cues as well as the nutrition. So thus these phycobilisomes, they can be modulated by availability of nutrients, light, etc. And we have actually exploited these factors for their enhanced production. Purity of these pigments is of utmost importance, as their intended application will depend upon its purity, as I've told you. And here you can see, although there are a number of methods which are available, but an economical method and with faster and higher recovery is something that may aid in their commercial production. So looking into that, we devised a one-step purification process using anion exchange chromatography. But before we really go for purification, we need to extract these pigments. And again, there are so many methods. Here we compared different methods of extraction using phosphate buffer, lysozyme, and acetate buffer. And you can see here that the acetate buffer, which we used of 20 millimolar acetate buffer of pH 5.1, of course, containing with this 0.05 molar NaCl and 0.002 molar sodium azide. This was used as a successful buffer for extraction, or I would say better extraction of these Phycobili proteins. And then once you dissolve the cells into this buffer, then it is followed by freeze thawing cycles. And this again needs to be optimized from organism to organism. In fact, the number of freeze thaw cycles will vary with the kind of organisms, cell morphology, so that the maximum pigments can be taken out. This is, I would say, the schematic view of different purification steps, where we obtained purified phycocyanin and phycoerythrin. Initially, we have the ammonium sulfate precipitation, and once we get the precipitates, in fact, again here we go for fractional precipitation. Initially, 20% ammonium sulfate is used, which basically removes all the cell debris as well as contaminating proteins. And then after that, it is followed with 35% ammonium sulfate. And this 35% ammonium sulfate precipitation results in most of the phycoerythrin being precipitated. And then finally, we go for 65% ammonium sulfate precipitation, which basically precipitates all other Phycobili proteins. And then this is followed by the dialysis step, which is important, where all the salts need to be removed, and this is done with the 20 millimolar phosphate buffer at pH 7, and this dialysis is done overnight. In the demo clip, you will see that how we are going for this whole process. And then we go for the final or the step where we go for anion exchange chromatography. And before we go for this anion exchange chromatography, the column needs to be packed, and the column is packed with using the anion exchange resin, that is DEA cellulose 52 resin, which we are using here. And the column is equilibrated with the 20 millimolar phosphate buffer. And we have a gradient of NaCl, which is done here. And then you can see here these on the left down corner, these fractions are collected, and you can see the phycocyanin comes out first, followed by phycoerythrin, the pink color, and very nicely the pure fractions are obtained by this method. And this particular method we have validated in many of these cyanobacteria, and we could obtain, you can see here, stepwise purification. We could obtain a very good purity of above 3 or 5 or even 6.76 purity we have obtained in a couple of cultures, and the recovery is also good, which is beyond 40% after the final step. And this particular process, we have filed a patent, which is at the last stage of, I would say, awarding. All the examinations have been done for that. And you can see here the purity profile of nostoc species. I shall be concentrating during my lecture on two main organisms, nostoc and anabina, where we have found these pigments in a very good amount. And you can see here the step 1 is the crude extract, step 2 is ammonium sulfate precipitation, step 3 dialysis, and then it is followed by anion exchange chromatography. And you can see here as we move, the phycocyanin comes out first, and then phycocyanin amount is reduced, and phycoerythrin is dominating in the next of the fractions. So that is how we basically obtain or fractionate these Phycobili proteins. And this is from Anabina variabilis. Again, you see here it is having a very good amount of phycoerythrin. Well, we have also basically gone to did some calculations for this simple method which we devised. Of course, this is not a very realistic estimate, but the cost we got was about 1/10 of the cost for which it is sold by Sigma Aldrich. 1 milligram of C-phycocyanin of purity more than 3.5 is sold by Sigma Aldrich for US dollar 158, which is in Indian rupees is coming around about more than 11,000 rupees. But as far as our calculations, we get 1 milligram of C-phycocyanin of purity more than, I would say, 4 for about 1,000 rupees. So this is our calculations which we have got here. And so about 1/10 of the cost we got this using this particular method. This is the characterization. We have characterized this spectrophotometrically with using STS page as well as with HPLC. This is the absorption spectrum of phycocyanin and phycoerythrin purified from nostoc species as well as Anabaena variabilis. And you can see here a very good peak at the lambda maxima of phycocyanin, that is 620 nanometers, and of phycoerythrin, which is at 570 nanometers. Similarly, this is the STS page analysis of purified phycocyanin and phycoerythrin, and you can see here, depending upon the organism, there is a little variation in the molecular weight. You have the phycocyanin from nostoc and Anabaena variabilis having a similar kind of molecular weights for alpha and beta subunits, whereas from Anabaena oryzae, the alpha unit is 19 kilodaltons, but the beta unit is similarly 17 kilodaltons. Similarly, the phycoerythrin is alpha unit 18 kilodaltons and beta unit 14 kilodaltons. So all this has been characterized. We also did the identification of these subunits using MALDI-TOF MSMS, and we could see that our subunit of nostoc commune was resembling that of Microcystis aeruginosa. And what I would like to mention here, that the theoretical molecular weight and the actual molecular weight, they were very close to each other. And similarly for Anabaena oryzae, which was matching the nostoc species PCC 7120. Again, we characterized it with using HPLC, and why I have mentioned here all these things, because here the C5 column, we have characterized this or analyzed this using C5 column for the first time. Whatever earlier reports were there, they were using C18 column or C30 column. We used C5 column, and we did use detectors of PDA as well as the fluorescence detector, and we got very good, you can see here nostoc species, the PDA detector at 620 nanometers, very good two prominent peaks of alpha and beta subunits. And when this was done at 226 nanometers, again we got two peaks at 27.32 minutes and 28.294 minutes corresponding to alpha and beta subunit. And the fluorescence detector, when it was set at excitation of 580 nanometers and emission of 640 nanometers, again we got very good alpha and beta subunits of the purified phycocyanin. Similarly, we did it for, and this was the spectral characterization of alpha and beta subunits. The ratio of A280 to A640 was 1, and this showed the presence of one phycocyanobulin as chromophore. And in the beta subunit, this ratio was 2, showing that there were 2 phycocyanobulin chromophores present there. Similarly, it has been done for Anabaena variabilis. Again, the spectral characterization for this similar kind of results we got. And then we took this purified and lyophilized sample, and we did infrared spectrum of purified phycocyanin, and we could get the peaks for these all these bonds, CO stretching bonds, NH bending, and this CH bending aromatic ring, which was below 800 centimeters. So that shows that the purified phycocyanin was stable, and all these bonds were intact in both nostoc and Anabaena, where we carried out this study. Then, as I told you, that all these are basically regulated by different nutrient components as well as the physical conditions. So we studied the production and regulation under different conditions. Although we have detailed studies, but in nutshell, I am just presenting here. You can see here the gel on the right side is about the effect of iron sources as well as iron concentrations. We used 3 irons. On the left hand side, you can see the phycocyanin production, and in fact this regulation we saw on phycocyanin gene, CPCB gene, which is responsible for beta subunit. And we studied the expression of this gene, and we took different iron sources, you can say ferric ammonium citrate as well as ferric chloride and ferrous sulfate. And what we could see that ferric ammonium, the production was more under ferric ammonium citrate was used as a iron source. And also you can see here the expression was much better under ferric ammonium citrate, whereas it was less and when different other sources were given. And what we could see that 1.2 ppm of ferric ammonium citrate was the best concentration for better expression of this particular gene. Similarly, we took different light intensities of 55 micromole photons, 35 micromole photons, 45 and 65, and you can see the variation in the expression of the gene under these conditions as well as production. Of course, we had a mixed results here where a lower light intensity could also lead to higher production, whereas higher light intensity also led to better expression of this. And these are basically very well modulated by light intensity as well as light quality. I have a couple of slides where light quality effect on these Phycobili protein production will also be shown. Similarly, we studied the salt concentration. We had different salt concentrations ranging from 0.01 molar to 0.1 molar. And what we could see here that a little salt stress of 0.01 molar could increase the expression of this CPCB gene in Anabaena variabilis. This is how one can enhance the production of these pigments by using these metabolic modulation, I would say. This is again a similar kind of thing which we did for nostoc species, and here we could also see that ferric ammonium citrate was better represented, and in case of light there was a better production, you can say, under higher light intensity. And for salt, of course, nostoc was not able to grow at 0.1 molar. So again, 0.01 molar concentration was better for better production of Phycobili proteins. And this is what I was talking about, effect of light quality on biosynthesis of these PBPs. And you can see here a clear-cut differences when the white light was given, there was more production of phycoerythrin. But if you go to the right side below, blue light, you can see it is actually reversed. Under blue light, there is more production of phycoerythrin as compared to phycocyanin. And of course, this is only one slide I am showing here. But normally what has been seen that under white light, we could get better biomass, but under blue light, the Phycobili protein production was more. Similarly, in Anabaena variabilis, you can see in white light, again, the phycoerythrin production is more. And as we go to blue light, not much differences are there, but more or less similar kind of. So it also varies from organism to organism, but one can very well modulate and you can see here under red light, there is a total production of phycocyanin, and the production of allophycocyanin is 0%, and phycoerythrin is very negligible. So if one wishes to go for exclusive production of a particular kind of Phycobili protein, one can choose a different kind of a light quality, but then again the organism also matters. Then once we basically were sure, or we saw that all these media components they are affecting the production of these PBPs. So in fact, our aim was to enhance the productivity of these pigments by all these biochemical modulations. And as is always done, so initially we used response surface methodology to optimize PPP production in, as you see here, in Anabaena variabilis. And here you can see that we took these three factor and one response experiment where 20 different experimental runs were there. And what we could see that maximum Phycobili proteins of the tune of 0.39 milligram per ml, they were obtained with the predicted optimal levels of these three input factors, that is ferric ammonium citrate at 0.12 grams and K2HPO4 0.05 grams and trace metal of 0.5 ml per liter. So these are the ranges which we did select for this particular experiment. And you can see here, these are the three-dimensional interaction graphs and showing interaction of all these input factors. And of course, the results showed a quadratic effect of dipotassium hydrogen phosphate and linear effect of ferric ammonium citrate and trace metals on Phycobili protein production. But then what we did, after this response surface methodology, this was followed by a new, I would say, statistical approach of optimization of Phycobili proteins production in Anabina variabilis using genetic algorithm and fuzzy logic methodology. And this fuzzy model or fuzzy sets, they have the ability of recognizing, meaning, controlling, deciphering, and using data having absence of assurance and equality. So these fuzzy rules are created according to the connected relationship between input factors, output response values, and the interface activity has been connected with these rules. In fact, these 5 membership Gaussian functions, they were considered for each input parameter, that is ferric ammonium citrate, K2PHO4, and trace metals, which we took for RSM. And the response factor, that is Phycobili protein production, was further divided into 9 levels. And that is some of, I would say, 20 different rules were created by considering the maximum minimum compositional procedure. And the outcome of these fuzzy logic model was compared with experimental data to validate the designed fuzzy model. And these you see on the top, these three-dimensional surface interaction plots of input factors and the convergence plot of Phycobili proteins production fitness value with the number of generations. And what we could see here, that the productivity of Phycobili proteins was enhanced by about more than 2 folds over control by changing the optimum these combinations of selected factors. And you can see here that the total Phycobili proteins under non-optimized conditions was 190 milligram per liter. With RSM, we got 385 milligram per liter, but by applying this genetic algorithm with fuzzy logic methodology, we still got a better production of 405.20 milligram per liter. And this has been validated experimentally, and this particular thing we published in Bioresource Technology. Similarly, for another Nostoc, we again carried out the response surface methodology. As you see here, this is the whole, I would say, summarized statistical optimization which we carried out for Nostoc using connected neural network integrated with multi-objective genetic algorithm, what we call as CNN MOGA modeling. And so here we initially did the RSM where we used 4 factors along with a multi-objective response. That is, it was not only Phycobili proteins, it was biomass also. And then we developed the connected neural network, and the best connected neural network model was fed into this multi-objective genetic algorithm tools to predict these input factors. And then experimental validation was done. And you can see here, this is the RSM which we did, and these are the 4 experimental inputs. Again, ferric ammonium citrate, magnesium sulfate, K2HPO4, and pH these four were taken, and these are the ranges which were there. And with RSM, again, we got very good results of enhancement from 210 microgram to 309 microgram per ml. And there was also a biomass enhancement of about nearly double, but R square value was little less. But then this was followed by, as you see here, the multiple objective optimization using connected neural network and genetic algorithm. And the biggest advantages of multiple objective optimization is that it provides optimal solution for more than one objective simultaneously. Here we have two objectives, the biomass production as well as Phycobili protein production. So a hybrid evolutionary algorithm, what we call as CNNGA tool, was developed to investigate the optimal solution for input parameters for maximizing Phycobili protein production along with cell growth. And this we did in Nostock. And the hybrid evolutionary algorithm was run continuously to obtain optimum input values of selected input parameters, which, as I've told you, they are the components of BG11 medium. And this was run in MATLAB 16 version of this particular software. And basically, for RSM, we run about 30 different runs, and out of those 30 experimental sets, 21 were used for training, 5 were used for testing, and 4 for validation, as has been shown here. And we obtained these regression plots by this. And the R value which we obtained here was 0.99518, which means that 99.52% accuracy was there. And the R value for others are also, you can see in everyone, it is near to 1, which is basically very good for this. And this is the Pareto front graph, which was basically, as you see, saw the 3D interaction graphs earlier. But here, because only two objectives are there, objective 1 and objective 2, so we selected the linear graph. And you can see here that by using CNN MOGA, we got a very good enhanced production as compared to RSM. This was 340 microgram per ml. So these kind of statistical near human approaches have been used for the first time for biopigment production in cyanobacteria. And this particular work on Nostock has also been published only a couple of days back in, again, Bioresource Technology journal. Now, once we optimized these production schedules, then we tried to upscale this using different modes of photobioreactor, which is of 3.5 liter capacity with us, and then also in trays of about 18 liter capacity. And you can see here, and we selected Nostock for this purpose. And you can see here that, of course, the biomass production was more or less similar till 35 days in both photobioreactor and tray. Of course, in photobioreactor, there was a little enhancement after 20 days. But then if you see the Phycobili proteins production, after 20 days in photobioreactor, there was enhancement of Phycobili protein production as compared to its production in open trays. Of course, these open trays were lodged in the glass house. And so there was much better production of Phycobili proteins under controlled conditions as compared to the semi-controlled conditions in open trays. And then we fractionated these Phycobili proteins in photobioreactor, and you can see here that the fractionation of these revealed that phycoerythrin production was more, followed by phycocyanin and allophycocyanin. And of course, allophycocyanin amount was negligible as compared to the phycoerythrin and phycocyanin. Then in order to further economize the large scale production or the pilot scale production, we devised different kind of media using some cheap nutrient sources, and we in fact devised 5 different kind of media. And you can see here we compare it with the traditional BG11 medium, which is used for the cultivation of cyanobacteria, and you can see that the Phycobili protein production is statistically on par as compared to its production in BG11 medium when we have used medium 3 and medium 5, and the production was carried till 35 days. Then, if you remember, we had studied that a small amount of salt stress could enhance the production of Phycobili proteins. So this we also tested under photobioreactor conditions, and what we could see here that with the use of 10 millimolar NaCl in the medium, the total Phycobilin pigment production increased by about more than 12%, 12.78% to be precise under salt stress, and maximum production could be achieved by 30 days. So this fractionation of phycobilins again showed the dominance of phycoerythrin and phycocyanin. Of course, we have carried out many more studies with upscaling, going up to raceways also, but those results I am not showing here because of the paucity of time. So I would conclude my presentation by saying that, of course, everybody knows that these pigments, the Phycobili proteins from cyanobacteria, are very valuable and have a lot of applications. So we have devised an efficient and economical method for purifying these phycocyanin and phycoerythrin from different cyanobacteria. The expression studies have shown regulation of CPC-B gene through light intensity, iron, and salt stress. And the Phycobili protein production has been very well or successfully optimized in Anabaena variabilis and nostoc species using response surface methodology, followed by the statistical approaches of genetic algorithm and fuzzy logic methodology, as well as multi-objective CNN genetic algorithm models. And the productivity of these PBPs was enhanced by more than 2 folds. And upscaling of the biomass and Phycobili protein production has again been optimized in photobioreactor as well as in open trays. And we could get a better or enhanced production when we have used NaCl at a lower concentration of 10 millimolar in the photobioreactor. I would like to acknowledge my collaborator, Professor Pratyush Shukla, who has now joined School of Biotechnology at Varanasi, and my students, Dr. Hilol Chakdar, who is now Senior Scientist at the National Bureau of Agriculturally Important Microorganisms, and Mr. Dinesh Kumar Saini. Both these, they worked on biopigments, and Dinesh has also submitted his thesis. There were some questions asked by the participants. Of course, all these questions are not much related to the today's topic. However, because we have been working on different aspects of cyanobacteria as well as microalgae, so I would like to briefly answer these questions. The first question is how we can exploit microalgae in efficient nitrogen fixation in agriculture sector. By Mr. Isaac Russell. Mr. Russell, if I mention here that microalgae, especially cyanobacteria, they have been extensively used as biofertilizer in rice-based cropping system, and it has been since the time, I would say 1940s, when it was hypothesized that the fertility of rice field soils is due to the presence of blue-green algae. There has been a lot of work going on the use of cyanobacteria as biofertilizer. especially in the Southeastern Asian countries. And we have at our center developed and produced these biofertilizers, BJ biofertilizers, and we are giving it to the farmers. And because these cyanobacteria, they fix atmospheric nitrogen, so the primary contribution is of nitrogen, and it is actually said that 25 to 30 kilogram nitrogen per hectare per season per crop is provided by inoculation with cyanobacteria. Besides that, there are many other, I would say, benefits of using cyanobacteria. They improve the soil, they provide growth promoting substances, and not many things are there. But as a layman, one can say, because these are nitrogen fixers, so they provide nitrogen. So the chemical nitrogen input to the tune of 25 to 30 kilogram per hectare can be reduced by use of these cyanobacteria, and these are extensively used for this purpose. Another question is on characterization of microalgae to study if any bioremediation process is taking extracellularly or intracellularly. I would like to say that Ms. Sami, this bioremediation by microalgae is a multifaceted approach, and there are a number of mechanisms involved. Absorption is also there, adsorption is also there, and a good absorption is there because of the better surface is to volume, surface area is to volume ratio of these minute, I would say, creatures. Of course, transformation we have studied. Many of the metals which are toxic at a particular level, they are transformed or they are changed into non-toxic levels, especially the chromium, arsenic, which we have studied. And then many of these, I would say, the sources, nutrient sources, the nitrogen, phosphorus, all these, they are very well assimilated, which basically cause pollution of sewage or domestic waters. And then biodegradation of many of these dyes, as well as many I would say recalcitrant compounds, is reported by microalgae. So there has been a lot of work. Of course, application may be very limited, but lot of successful reports are there about bioremediation, and many of these mechanisms are there. And depending upon mechanism, one can study the whether it is, suppose you are studying the cellular biomass. If the products are there, that means it is getting degraded. If the similar, suppose same product or same compound is there, that means it is getting adsorbed or absorbed. So all these mechanisms can be used for their analysis as well as characterization. Then another question by Dr. Noreen Khan is about how can we use the microalgae in cost-effective removal of recalcitrant compounds present in water bodies and as a source of energy to carry out different domestic works, also in minimizing air pollution. So it is more or less similar to the earlier question. Of course, microalgae can be cultivated in all these waste and polluted waters. And since these microalgae, they use CO2, so they sequester CO2, they improve the environment of all these greenhouse gases, and they release oxygen in turn. And then the biomass which is produced, that can be used for energy or biodiesel production or many other related aspects, which is extensively worked out in many of the labs. Then Dr. Raja Rathinam Ravi Kumar has asked why phycocyanin is not having much market value than spirulina. My dear Dr. Raja Rathinam, it's not true. Phycocyanin has a great market, may not be in India, but globally the phycocyanin is produced by many companies, and many of these products are available. As I said during my lecture, that it is based on the purity. Of course, for food, maybe people are not preferring to use these natural colors, because maybe we need to see the economics of this, but depending upon their purity, they have been used for many analytical applications, the pharmaceuticals, as well as now there is a big market for cosmeceuticals because of the antioxidant and immunoregulation properties of phycocyanin. And of course, spirulina as a biomass is extensively used. So many of the companies, they are producing spirulina and selling it, because for phycocyanin one needs to go for processing and infrastructure required, and setting up of all those I would say different kind of equipments and apparatus requires some investment. So that's why spirulina has a better market, but then phycocyanin has its own I would applications which are very much important. Then lastly, is there any microalgae which can be utilized as a biocontrol agent to manage plant parasitic nematodes? Yes, a number of microalgae have been tried, especially I would say even in IARI, the nematology division, we are having a collaboration where Cyanocystis, Anabina, all these have been successfully I would say seen or tested for control of especially root knot nematodes. And there are reports where macroalgae, the seaweed extracts, they have also been extensively, I would say, tested for control of nematodes. And because of the presence of these phenolics and many other enzymes which are there, especially proteases, which are able to control these nematodes. So if you want, anyone wants some details about these aspects, or otherwise about the Phycobili proteins, which I have discussed in detail today, so you are most welcome. The organizers have all the details and contacts of our lab, so any one of you is most welcome to discuss with us. So thank you very much, and once again I would thank the organizers for this giving me time.
Dr. Papi, so shall we play your video clip?
Yes, I think you have with you.
Yeah, we can play at our end.
So I will.
We are here demonstrating a process for laboratory scale production, extraction and purification of Phycobili protein pigments from cyanobacteria. The foremost requirement is to have the cyanobacterial culture. The culture is cultivated in specific medium, say BG11 medium, which we are using here, and this can be with or without nitrogen depending upon the species. Initially, inoculum preparation is carried out and log phase culture is inoculated in the fresh medium. Normally we use 2% inoculum, and the whole process as shown here is performed under aseptic conditions in a laminar flow so as to avoid any contamination. And once this inoculation is complete, these culture flasks, they are then incubated in a growth or culture room under controlled conditions of temperature and light as shown here, 28 degrees Celsius plus minus 2 degrees Celsius, and light intensity of about 65 micromole photon per meter square per hour, and with 16 to 8 hour light dark regime. And one can incubate these for about 21 to 28 days so that these cultures attain full growth. And once these cultures they attain full growth, and you can also basically shake them manually 3 to 4 times a day so that they don't form any clumps or they don't stick to the walls. And once these are fully grown, then they can be taken for the next process. And And here I am showing that instead of culture room, one can also use an incubator with light and temperature control facilities, and such kind of incubators can be used for growth of these cultures. And once these cultures are ready, they are basically harvested, and after that the culture is first homogenized properly, and after homogenization it is centrifuged at 3000 rpm for about 5 minutes, and the cell pellet which is formed is washed twice with distilled water, and then it is suspended in 20 millimolar acetate buffer of pH 5.1 and then subjected to freezing and thawing cycles. And these freezing and thawing cycles, they result in rupture of these cells and the pigment comes out. And you can see here very good pigment is shown here in the beaker. After extraction, the purification is done. And the first step in purification is salt precipitation using ammonium sulfate. And for better purification and maximum recovery, fractional precipitation is performed. Initially, 20% ammonium sulfate is used to eliminate any cell debris as well as contaminating proteins. And the solution is this ammonium sulfate is put in the pigment solution. It is properly dissolved, and after that the precipitate which is formed is basically centrifuged at higher speed of 10,000 G for 15 minutes at 4 degrees Celsius. And then the supernatant which is obtained is subjected to 35% ammonium sulfate precipitation, which results in precipitation of phycoerythrin. And finally 65% ammonium sulfate is used, which causes complete precipitation of all the Phycobili proteins. And now dialysis is carried out, and this process of dialysis is carried out at 4 degrees Celsius to prevent any damage to the protein pigment. The process takes about 24 hours, and next day the sample is centrifuged at 10,000 G for 10 minutes at 4 degrees Celsius, and the supernatant which is obtained is containing the pigment which is free of any cell debris. And this particular pigment which is there, it is further basically purified by passing it through micrometer filter. The pigment is taken in a syringe and then it is passed through a micrometer filter, 0.45 micron filter, and this pigment which is obtained is basically subjected to the final step of purification is anion exchange chromatography. Here you see that the column is packed with activated DEAE cellulose 52 resin. The dialyzed sample is loaded on the column and allowed to bind with the resin material. Care should be taken to load the sample as a discrete band so that elution is proper. Elution is carried out with 20 millimolar phosphate buffer using gradient concentration of sodium chloride, which ranges from 0.02 to 0.2 molar. And you can see here that once this pigment is loaded, and you can see that phycoerythrin comes out first, followed by phycocyanin, and then these eluents or these fractions, they are collected in different tubes, and about 5 ml of each fraction is collected. And you can see here that the pigment is coming out from the column, and these different fractions, they are collected in separate tubes. And once these all fractions are collected, and you can see here the first couple of fractions, they are pink in color, they are having phycoerythrin, whereas this is followed by phycocyanin. And then these fractions, they are basically characterized and analyzed using UV visible spectrophotometer, and here the scanning is done over a wavelength range of 200 to 800 nanometers, and this pigment is put in the cuvette and cuvette is there in the cell holder, and then it is basically scanned from 200 nanometers to 800 nanometers to see the purity as well as quantity of the pigment. And you will see here the process has already been started, and the observations will be noted onto the monitor of this computer. And you can see here the scanning has already started from 800 nanometers. It will go up to 200 nanometers, and hundreds of these observations are being taken where all the absorbance is noted. And you can see here now the graph is coming here, and now because the absorbance was set at a lower range, now it is set at a higher range of 1.5, and you can see a very good peak of phycocyanin at 620 nanometers, and then it goes down, and further scanning is going on, and you can see that a very good peak is observed because all these pigments, they have a lambda maxima, and phycocyanin has a lambda maxima at 620 nanometers, so it has shown a very good peak at 620 nanometers. Similarly, phycoerythrin has a lambda maxima at a different wavelength, so accordingly it will show, and you can see here the purity of these pigments. So this is the whole process of basically production, extraction and purification of these Phycobili protein pigments from cyanobacteria, which is shown here. And this is a very simple two-step process which is basically developed in our lab, and we have also filed a patent for this particular process. I thank all my collaborators as well as my students and Mr. Ajit, who have helped in basically in preparation of this particular demonstration unit.
Thank you, Dr. Papi. We appreciate your active exchange of ideas and view on how we could efficiently address and fill in the gaps in the extraction of the phycocyanates from algae and their key challenges in the commercialization. So due to running short of time, so the participants can put their questions in the question answer box, and we will ask speakers to revert to you shortly. So we
thank you
move to
very
our
much.
next speaker.
So thank you so much.
So yeah, moving towards another interesting lecture from Dr. Nirupama Malik on B3 approach, biomass, biofuels and biorenewables from cyanobacteria. I would like to invite her, but yeah. Dr. Nirupama Malik holds the position of Professor in Agriculture and Food Engineering Department, Indian Institute of Technology, Kharagpur, India. She is an algal biotechnologist and working on various aspects of algae and cyanobacteria for more than 3 decades. Professor Malik's research career began at Department of Botany, BHU Varanasi, which is also the Center for Advanced Study in Algology. Later, she moved to Germany as an Alexander von Humboldt Fellow. She has contributed well over 100 research papers in various scientific journals of international repute, and several book chapters and reviews related to various aspects of algal and cyanobacterial physiology, biochemistry, and biotechnology. She is working in the area of cyanobacterial biorefinery for more than 3 decades. She is also fellow of the National Academy of Sciences, India and Indian National Science Academy. So I request Dr. Nirupama to start with her lecture.
Thank you, Ruchi. But we are a little late, so how we can accommodate, or I will go on about 20 minutes. So I am sharing.
Yeah, you can go on, ma'am. Yeah. We'll see. We can take the questions answers in the after the session, so we can compensate with that.
Okay.
You can continue, ma'am, please. Ma'am, if you find any issue, we can share the presentation.
No problem. It is here. I am sharing. Is it visible?
Yeah.
Yes, yes.
Perfectly visible. Yeah.
Okay, a very good afternoon to all the participants, and I will try to manage a little bit of time because my designated time is up to 1 p.m. But maybe I will take a little bit more time because of this session running late. So let us start it like that. We are moving towards the era of biofuels, and as we are knowing that, particularly for the student participant, I am telling, if you only look at the biofuel part of the microalgae or cyanobacteria, particularly microalgae, as we are considering, mostly it is considered that we focus on microalgae and increase its lipid content, and then go for biodiesel production or biofuel production. Particularly biodiesel we are focusing. But there is another source, you can say, that is cyanobacteria, and we all know them because they are very good nitrogen fixer, and from our college time we know that cyanobacteria are famous for their nitrogen fixing ability, and they have a specific cell that is called heterocyst, that all of you know. But they are also a resource of many chemicals, you can say, or many products, and that is what we are focusing for in our lab is focusing for. So only focusing for biodiesel or biofuel, that is biodiesel or bioethanol, we cannot, there is always a cost factor. So we have to valorize the whole biomass. So to get more and more product, like Professor Papi has told now, the cyanobacteria is a very good source for phycocyanin, and phycocyanin has many aspects. You can say that crude phycocyanin you can use for natural food colorant, and then also they can be used in fluorescent tag in radioimmunoassay, and there are many aspects, like they have antioxidant properties, medicinal values, many things are there. But mainly it is not yet explored, you can say. What I have heard from the question and answer session also, people are telling about some questions are there on that why spirulina has a market and this one has not. Because we have not come up to that label, that we are not exploring these organisms, and we are leaving them. Spirulina is known for protein product, and but all these cyanobacteria, you can say, they have many they contain many compounds which are not explored. Okay, if you see that we have a very good product, well-known product for all our student participants, that is agar. Agar we all are using, but that is also a product from microalgae. Okay, so as I am going through this B3 approach, that is biomass, biofuel, and biorenewable, that means if we don't get much biomass, whatever product we are focusing will not get sufficient quantity. That is a productivity related issue. So biomass should be high, then we can get biofuel or whatever product we want. And then the biorenewables, why I am telling, we have to try to valorize or extract many products from the same biomass. That is as a refinery concept. So we may reach up to a level where we can become the cost competitive. So the market value will come. So now here what we are focusing, as you can say, our lab is also working on algal biodiesel from many years, and with cyanobacterial different product for you can say many years. But here we are focusing not the biodiesel but the bioethanol part. And cyanobacteria, you know, they are a very good source of carbohydrate, and they grow very well in wastewater also, as you know. And these cyanobacteria, they have the glycogen as their storage carbohydrate. And glycogen is nothing, it is a polymer of glucose. So it is a very good source for fermentation by microorganism. So what we are working to try to focus on the carbohydrate, and then the how to produce from that the carbohydrate, the bioethanol, then also how we can get some other products under a refinery, biorefinery concept. So this is what our main focus of the study is. We have cyanobacterial biomass. Maybe the inherent carbohydrate you will find not enough, or whatever it may be, there will be a basic level. Then we need some strategy to enhance the carbohydrate. So then we can think of increased bioethanol production and also various co-product. That is, then we can got some expected under the refinery, biorefinery concept. So we can get some expected rise in the production cost, and so, or we can get this production cost of this bioethanol can be adjusted. So now also we can say that cyanobacteria is a very good group of organism. They are capable of these nutrients from wastewater. There are many reports you can find out that I have also wrote a review on this, that how by immobilized cyanobacteria only, and so that is there, and a lot of citation to that, and they are capable of your taking the nitrogen, the phosphorus, and also organic matter from the waste. So they can also bioremediate the waste, and simultaneously you generate the biomass, and that biomass can go for further processing. So that is also one aspect. And now what our lab is focusing on this part of the study, I am coming to, that we have some many cyanobacterial species. So we first try to find out what is the proximate analysis, because we need an organism which is a very fast grower when we are thinking of the biomass, and also it's the carbohydrate content should be high. So among them, we first select a cyanobacterial species. This is, you can say, experimental design, which I want to share with all the students. And this is then you find out the total carbohydrate production and how we can improve with the carbohydrate productions. There are some reported parameters or variables, you can say. You give nutrient starvation, and normally nitrogen and phosphorus, they are given as stress or deprivation. So there is a increase in the production or accumulation of your desired compound. So now, but these are, you know, they are, if you see the growth medium, these two are major nutrients. So when we deprive this nutrient, so growth is very low, and so therefore you cannot, when growth is low, so overall it will affect the biomass production and then the production of your desired compound. That is what we have taken this to see how it is happening and how it is, what it is doing with the carbohydrate content of the organism. Then these four compound or four variables which we have which we have identified, very simple experiments we have done. That is, we followed the Lellinger book, Lellinger biochemistry, and for biomass production, photosynthesis rate should be high. And if you read that photosynthesis chapter, you will find the pH, the magnesium supplementation, calcium supplementation, and bicarbonate. You can say these things, they affect the photosynthesis. So overall the biomass yield. So, and also you will find they have specific role in carbohydrate accumulation. So we follow that and then we identified some variables from here, the important variables, and then we optimize it that to get the interaction of multi-factor, how they are affecting. Then after that, we see the how the carbohydrate yield. Then from the carbohydrate yield, how we can go for bioethanol production. And this carbohydrate also profiled for reducing sugar, glycogen, starch, cellulose, and hemicellulose. And also, as a refinery concept, we try to find out co-products, various co-products. There is one is C-phycocyanin. Our laboratory also developed a very simple method how we can extract the phycocyanin that we have also applied for a patent, and that usually we show in our MTech lab, and a very simple method. Then for polyhydroxy butyrate, this is this you may know that there are these are one type of biodegradable polymer or plastic, you can say. Our lab is also working for many years in this one with cyanobacterial PHB. Then SCC. SCC is sodium copper chlorophyllene. This is nothing. This is a you can say this is a water soluble component or water soluble component of your chlorophyll. And because if you, what we are doing, that is standard method, if you replace the magnesium ion with copper, you can get this one, and that is, this is water soluble one. So it can be used as a natural food colorant. Then this is EPS. EPS is your eco polysaccharide. This eco polysaccharide, you see cyanobacteria when they are growing, they have a bloom, they form a bloom. And how they are, there is some mucilage is always in their secretion. That is what we are focusing also. That is called the polysaccharide, and that is also have good. This also has a good market. So these four products we are focusing in this study. So among them, we have identified one or selected one organism. You can say this is Anabaena variabilis. And with Anabaena variabilis, we have tried with all these experiments, and you can say we have checked with the control. If you see here, this is the biomass yield, and this is the carbohydrate yield, and this is the percentage. That is the 46% is carbohydrate, total carbohydrate. And with phosphorus and nitrogen starvation, you can say phosphorus starvation is very good. It is increasing the content or accumulation to 61%. But if you see the biomass, you can imagine here that it is it was reduced to less than even half. So overall, if you see the yield, our yield is less. So this is, if you target this one, we cannot get a, because our main focus is how to get more and more carbohydrate, so we can convert it to bioethanol. Okay. So now if you see this, then this is these two factors we cannot take, and with the culture pH, we have tried many experiments. Also with magnesium, different concentration, calcium also different concentration, and bicarbonate. So these three major factors which we have found, they are increasing the yield of carbohydrate, our main target product, is very, very high. So then we optimize this with applying this response surface methodology. That is a statistical package. If you get that, you get a number of experiment it will give you, that is 20 experiment, and they all these three factors now they interact with each other, and we took a range like our whatever we have got in our single factor experimentation, that we have taken at the 0 level. Then we vary this to 2 level, you can say, and we got 20 experiment, and this 20 experiment we tried, and with that optimized, we got the biomass yield was increased by 1.5 fold, but due to accumulation of more and more carbohydrate, the carbohydrate was increased by 2.4 fold. So so this is a very good, you can say, yield for our experimentation. And then we focus that how these carbohydrate profiling, that about the reducing sugar and then glycogen, these two are very much very good for, you can say, your fermentation process. Then starch also, and then little bit of starch, you can say, cellulose, hemicellulose, they are not very good source for fermentation. And you can see from here the yield of all these compound, these two major compounds, so which are focusing, they are also like reducing sugar and glycogen, they increase significantly. Also there is a rise in starch content, but the hemicellulose, cellulose content was not so significantly rise. Now, as usual, we have gone for a pre-treatment of this carbohydrate, so that will make the bonds more, or break the bonds, so we can then it will be easily fermentable. So we have done that, and we selected with the 2 normal H2SO4 and with autoclaving, we can our maximum bioethanol conversion we are getting with that. And you can see from here we have the control, you can say our bioethanol yield is low, you can say, and but the conversion in control and the optimized conversion is same, that is around 45%. And here it is about more than 2-fold rise or in the bioethanol yield. And we also checked with the GCMS. There is a little bit water content also, but which will be taken out or separated. This is our ethanol peak, and this is the internal standard as 1 propanol, and this is the water. So this water can be eliminated with the fractional distillation in the large scale process. So now coming to different product, as I have told you, that is C-phycocyanin, then polybeta hydroxybutyrate, then sodium copper chlorophyllene, and also S-polysaccharide, that is EPS. The EPS, I am telling that it is it has 2 components. One is release polysaccharide, which you can get from the medium, and also one is this is the your CPS, that is the capsular. It is capsular polysaccharide. It make a you can say ring around the cell. So that also we try to get from the whole organism. Now you can see that is our C-phycocyanin. This is the freeze-dried C-phycocyanin, and this is the extracted C-phycocyanin. And under the optimized condition, there is a significant rise, and this as compared to control. And this is crude type. Professor Papi had shown that that is purified one. So it has a 1 peak, but it is a crude type. So it is a, if you study this, you will find that this is Ficobili protein. It has a protein linker there. So we have a peak at 620 and also the protein peak around 280. So this will be there. And if you go on purifying it, then gradually this protein part will be less and less. You can check in the Sigma catalog. There is a 40% purification and this percent purification, and how with that gradually the cost is increasing. Okay. Then now next is our PHB, which is extracted from the cyanobacteria Namina variabilis, and this is the confirmatory study. That is, this is the standard, and this is the sample, and we have a peak in the GC study, GC chromatogram, and this is the internal standard is your benzoic acid profile ester. And also under this optimized concern, you got a rise in the productivity. And also this is how this is the sodium copper chlorophyllene, where the magnesium ion of the chlorophyll was replaced by copper ion by several steps, and it makes this sodium copper chlorophyllene, and which is a water soluble green color, you can say, and you can use that as a natural food colorant, and it is very stable up to, we found it is very stable up to 90 degree and 90 degree centigrade, and in 100 degree it start degrading. Okay, so now this is all the confirmatory study of the standard and also the variability sample, and these are some of the confirmatory test for copper test, because copper is present there. Then with the flame, with the flame test, we confirm that is okay, copper is present there. And this spectrophotometric study, you can check the lambda max, and then you can say it has 2 peaks, and so it is fully valid. Then we have extracted this EPS, that is exopolysaccharides, and it has many values, like you can say acylating agent and other market is there, food, and you can say our for beauty product it can be used. And then this is the HPLC profile of this one, and this exopolysaccharide, and we have got 5 sugars in this one, that is ribose, xylose, and mannose, glucose, and galactose. So now under this optimized condition, what we are getting, that bioethanol yield was enhanced by 2.5-fold because of the increase in the carbohydrate accumulation. Then your CPC or phycocyanin and SCC, that is sodium copper chlorophyllene, they were increased by 3-fold, and PHB was increased by 2.4-fold, and EPS by 1.8-fold rise. So now the next part is when we are talking of a biorefinery. This is all individual component which were analyzed from the biomass. Okay. Now, when we are coming for the refinery concept, then which compound or which product will keep fast? That is also a challenge. So we can get maximum yield. So that is how we propose for a sequential processing approach for an cyanobacterial refinery. So we have selected, we put all our product in 6 different sequence, and we analyze that. And we found with this sequence 3, that is we got release polysaccharide, then SCC, then CPC, then this is your capsular polysaccharide, then PHB, and the last is carbohydrate, where this carbohydrate will go for our production of our bioethanol. Now we selected this one. Then we see, if you see with CPC, that means For release polysaccharide, which is in the medium, we can collect that and go for a exopolysaccharide market. But capsular polysaccharide, then we have several steps. We have to extract the capsular polysaccharide. So we thought that this is also a polysaccharide. So why we go so much for extraction of this? It is already the organism. Release polysaccharide are in the medium, so we can get it from the medium. But this one, why we'll go on extracting? We may use that for our main product, that is the bioethanol. So we leave this step or discard this step, and we make another sequence, that is we first go for RPS, then SCC, then CPC, then PHB, and carbohydrate for bioethanol. So there is a 1.4 fold, you can say 40% rise in bioethanol yield. Okay. So now this is our cyanobacterial proposed refinery with one main product, that is bioethanol, and 4 co-product. And that is what I told you, that ammonia variability culture. Then first we go for, we centrifuge and we took the biomass. And with the medium we got the EPS. Then we treat with acetone and we follow the protocols and got the chlorophyll extracted. This chlorophyll we converted it to SCC, and then we moved with the residual biomass. We collected CPC or your phycocyanin with distilled water. Then the residual biomass we gone for oven drying. Then we got the PHB or polybeta hydroxybutyrate. Then the third residual biomass we extract we have gone for acid hydrolysis part and then yeast fermentation to get the bioethanol. This is how our simple cyanobacterial refinery is designed with one student, and maybe gradually we'll add more and more product to this, and so the refinery will contain many components, and that is how we are going to make it cost competitive. And with this, you can say valorization of a biomass is 75%. That means 75% of the biomass was giving all these products, but still we have another 25%. Some will be lost, but we can try to get some more product from this 25%. So that will increase our refinery things. And this is actually a part of a work of one of my PhD students, that is Deepanita Deb, and she has published 2 papers, one in Journal of Cleaner Production and another is on renewable energy. So detail of all these and some cost analysis also was given there. Students can get it from there, and that will be helpful if they are interested. And she also has another part which I have not put because of this limited time, and that is how she focus on the various type of waste, and then we tried with this refinery concept, or we applied this refinery concept to this one. To the waste grown biomass. That is another type of study, and this is all. And thank you. If time permits, I will be happy to get some questions. And
so we have 2 questions from 2 of the participants. The first one is from Shailendra Singh. And the question is, what is the biochemical role of magnesium, calcium and bicarbonate in algae culturing, and how they can control carbohydrate biosynthesis?
Yes, this one you can say, this calcium, magnesium and your other things or bicarbonate, you can see pH and magnesium. they have several, their role is in several enzymes. Because you see, we normally grow cyanobacteria in BG11 medium, which has a pH of this standard pH is 7.4. But if you go higher pH, then normally photosynthesis and other things, they are much better. That is what, if you study Lellinger, you can get a very elaborative view of how pH is affecting, how magnesium ion is affecting. And all these are there because pH, calcium, bicarbonate is not there in the medium. Bicarbonate we added because of getting the knowledge from Lellinger, how bicarbonate is affecting. So several enzymes of the photosynthesis and you can say CO2 fixation, they are controlled by pH and magnesium. Calcium has several role in pH2 activity, very important role. If you study that, you will find in the oxygen evolution part, particularly you will see that. So then also bicarbonate, there is an enzyme called carbonic anhydrase, which is there that help in carbon concentrating mechanism for cyanobacteria. So you have to read all these things so you can get how carbohydrate mechanism, or we try to overall increase the photosynthetic efficiency, and these are found that they also increase the carbohydrate or sugar, because if you see our photosynthesis, the first product is our glucose. So how it is enhanced? So that is our, because we need more and more glucose, and so all these four things, they help, or they are very important component of photosynthesis. So very simple, you can say, from biochemistry, we have tried to get our desired product.
Okay, so next question is from Mr. Dharmen Shah, and the question is, can you suggest a methodology to find out components of RPS, component of the EPS?
Component of?
The question is the components of the RPS of the EPS.
Yes, the component are, you have to go for HPLC analysis because they are polysaccharides. So you could get the component by HPLC analysis. Okay, that is what we have. I have shown you some HPLC profile. Okay.
Now we can take one more question, which is from Chetan Paliwal, and the question is, what is the total lipid content in Anamena variabilis? Why this content is not evaluated in biorefinery approach?
No, lipid content, you can see overall the cyanobacterial lipid content is very low, 4 to 5%. We tried with that one. But if you see that the fundamental is that cyanobacteria has very low content of lipid. But microalgae, particularly the chlorophycean species, they have higher lipid content. And they respond. If you give them some type of stresses, their lipid content rises. But here it is not. Otherwise, we could have extracted the lipid and then convert it to biodiesel. Okay. Because the lipid content is very low, we didn't go for that. We also tried. You can see if they give nitrogen and phosphorus starvation to green algae, you will get a higher lipid accumulation, but not in case of cyanobacteria. We have tried that. So cyanobacteria is a different type of organism, and its behavior is different. It is prokaryotic nature and it is different. So we focus on when we are getting more than 70% of carbohydrate, and that is in the cost of your, you can say, lipid content is very low. That's why we didn't go for that. Okay.
Thank you very much for answering all the questions, ma'am, and for the knowledge-filled lecture and information on multiple products from cyanobacteria using D3 approach. So we'll move towards the last lecture, which is by Dr. Martin.
For the last lecture, I now invite Dr. Martin. Martin, a group leader at INL in Braga, Portugal. Martin got his PhD at the Material Science Institute of Madrid, Spain in 2011 for his work on self-assembly photonic structure for the light control at the nanoscale. After 5 years of research in University of Bristol, UK, He joined INL in 2017 to fund the Natural and Artificial Photonic Structure and Device Group. Throughout his career, Martin has produced seminal work on photonics aspects of photosynthesis, including the first demonstration of photosynthetic photonic crystal in plants. To this end, he has developed specific optical characterization techniques such as specialized microspectrometers suitable to determine the photonics property of microalgae in vivo. So I welcome Dr. Martin to deliver the lecture.
Thanks very much, Ruchi. One question: could I share my my own presentation, because I've changed a couple of things in one slide? So, if that's possible.
Yes, we'll share.
Okay. Okay, I hope you can see it properly now. Okay.
Yeah.
Okay, so yes, sorry.
Is it visible?
Yeah. Okay. Okay, so well, first of all, yeah, I have to thank you very much, all the organizers, Professor Alok, for the invitation, and it's really been a very nice workshop, and I've learned so much today. So, and thanks for the introduction, of course. So today I let me just put this in full screen, right? That will be better. Okay. So today I'm going to be presenting our work at INL. I mean, the work in my group at INL, and I've focused it a little bit on the characterization techniques we are developing or using related with microalgae, but obviously we have other topics as well, which I expect to go briefly through them. But before I get into the details, just let me introduce a little bit quickly where I'm coming from, or remotely coming from. Well, I work at INL, which is the International Iberian Nanotechnology Laboratory. It's a joint effort between Spain and Portugal, so the two countries in the southwest of Europe. No, we are the Iberian Peninsula there, and this is a very special place because it's an international institution, which means it has a very international environment, and by default is also joined by the two countries, no, which also give it this sort of multidisciplinary approach as well. And as you see here in all the topics we investigate at the institute, there are sustainable environment, clean energy, food-related research. So all of them connect very much with also with microalgae and other organisms, no, that are suitable for biofuel or for food production, for example. So this is very much aligned with today's workshop. And regarding my own group, well, we are sort of a very multidisciplinary group. So I'm a physicist by training, and I'm interested especially on how light interacts with nature, no? in general as a fundamental question. But I'm also very interested in how to use those materials produced by nature for specific applications, no? And as you can see here, so we are looking into photosynthesis and how nanomaterials inside the cells, nanostructures inside the cells can affect photosynthesis, therefore affecting the yield, for example, of crops, no? But we are also interested in learning those from those nanostructures and maybe using them as inspiration for actual devices. So at INL we have a strong device-oriented department. We have a big clean room, which gives us the possibility, for example, to learn how light is harvested inside the chloroplasts in the microalgae, and then try to mimic similar approaches, mixing wet lab and clean room technologies. And something specific for this, I will say for today's talk that might be of interest for several people is the use actually of the biomaterials as what we will call a naturally growth device somehow. No, I will show a little bit about this later because it's probably the part of our work which is more related with microalgae actually. And in fact, we have here a collaboration with TERI, and we submitted several proposals together with Professor Alok and Dr. Sobon Mandel. So, okay, so just let me introduce briefly the, let's say, more fundamental motivation, and I will go later to more to the technical motivations. But just to frame our work, I would like just to, so I don't need to recall here how photosynthesis works, like many of you know better than me, in the sense that that the chloroplast is for us like our unit somehow, no? And we are especially interested in the fact that light not only interacts at molecular level within the different pigments and protein complexes and so on, one can find in photosynthesis, but we are also interested on how light during the scattering within the cells will produce that the yield of that photosynthesis might be increased, might reduce. So this, for example, will come relevant similar to a photobioreactor. Depending on the height we are in the column, no, we get different absorptions. So here will be a similar approach, but we are looking at very small scales. And what happened is that when you look at very small scales, well, that light scattering might affect a lot. Like, for example, in this very nice paper by the group of Gregorkiewicz years ago, they show that just the stratification of the thylakoids might affect the way light is absorbed beyond the pigment content. No? But there are other examples, and these are maybe the ones that we are more focused on, are those where some intracellular bodies, in this case, for example, here we believe this might be some related with some lipids contents. So those intracellular bodies can generate nanostructures that can manipulate light themselves. No? And this might look a bit fundamental in the sense that they might not look very useful in the first instance. But if you consider, for example, in the case of this algae here, this is a brown algae, so it's not a microalgae, but for the case, it will help us explaining this. You can consider that this type of micromirrors that are formed by lipid spheres, so they are sitting on top of the chloroplast. So you can imagine that the filtering effect they are producing in the light, there is no pigment here, this is only lipids. And the reflectance is only produced by light interference. You can tell that that might affect how much light will reach the chloroplast. No? So this type of nanostructures, they turn out also to be dynamic actually. So we are in fact learning and even mimicking this type of structures, and we believe that we can use them in, for example, for pigments, non-dye containing pigments, no, in some applications. But apart from those biomimetic approaches, something which is maybe more interesting for in terms of microalgae or even biofuel production, no, is what are the functions of all these nanostructures in this type of organism? And there has been so many functionalities proposed for these so-called natural photonic structures, no, in the different organisms, that is very difficult to address a specific functionality for all of them. So if one considers high plants, that might be only just attracting pollinators. When in microalgae, as I will show later, we believe that effect might be actually related with photosynthesis itself, which brings me to the topic of the workshop, no? Which is, can we actually use, or can we learn something from this photonic approach to photosynthesis that then can impact in the biofuel production. But today I will focus on how we actually measure them. So measuring these properties is not straightforward in the sense that we need to measure in vivo. We need to measure we need to measure specific optical properties that are not related with the pigments, no? But since we need to do that in vivo, we have selected a specific organism, diatoms. We have selected diatom, the family of microalgae, the diatoms. And the reason for that is that they are obviously very important in terms of environment. Maybe they are not the most common microalgae yet in terms of biofuel, of course, even though their lipid content is high, but the upscaling of the production is not that that easy. But there is a specific property, which is the fact that they've got a silica exoskeleton. And the reason why that's very interesting for our approach is that this silica exoskeleton can work as a sort of optical filter. No, it can indeed produce a manipulation of the light at the nanoscale, and as one can expect, when light reaches the chloroplast, it has gone through all this nanostructuration, and it might suffer, for example, some sort of spectral filtering that might affect the yield of the photosynthesis, or at least the amount of light that reaches this chloroplast. But the other reason why we are really interested, and this is more on the technical side, is that indeed this biosilica has found many applications as a material itself, not only from the photonics perspective, not only from the how we manipulate light with them, but just as a porous, extremely porous biosilica material. So you can find plenty of literature on this, and as many of you might know, it has been already proposed as material even for battery, as anode for batteries in lithium batteries, for example, or of course applications in filtering is being used for blood filtering, for example. No? So we are interested in a specific topic of photonics, but of course, if we can upscale their production, the use of the biosilica will also be by itself will be a very interesting one. So, but how can we then study them? And not only the diatoms, this will apply to any other microalgae with photonic properties, but if we want to look at how light interacts with one of these diatoms in vivo, of course, we need to look at them in vivo because we need to know how they behave in their own environment. But we need techniques that are non-invasive in a way that we don't want to modify, for example, the quantum yield of photosynthesis during the measurement, right? But at the same time, we need single cell resolution because, as I will show you later, those properties are dependent on different morphological parameters. So we need to allocate those properties to a specific morphology, and therefore we need to go single cell for that. And finally, we need spectral resolution in our measurements. And this can get tricky as well, because as we saw very nice in this morning, in talk this morning, we can do, for example, confocal imaging in one of these cells, but usually at a specific wavelengths. No? So we need something more broad spectrally in order to analyze all these properties. And this is the type of techniques that we have been developing in the group. And just to give you an idea on where these techniques are, why these techniques are needed, I just want to remember that most of the techniques, when we analyze pigments, for example, as we saw this morning, we analyze in general, the absorption of those pigments, no, depending on the wavelength of the light we use to analyze. We just make a specific beam of light going through the sample, and we analyze the absorption in that particular pigment, and we can correlate that with the different pigment contents, no? But this has a lot of advantages, of course, and it has proved a very, very useful approach. But as I say before, in the particular cases we are interested, this will not apply, especially because we cannot extract, in many cases, we cannot extract those nanostructures from the cells. And therefore, this technique will not be useful for us. And there are other approaches that are used in vivo, and maybe the most interesting one for us will be the ones based in spectroradiometry, or if you want, for example, measuring reflectance, like in this case here, no? And we can still allocate from these reflectance measurements in vivo, still allocate specific features for each pigment. Now, the problem there is that we still need a biofilm. We are looking at many cells at the same time, not to get enough signal. So therefore, this won't work for us either. And then coming specific even more to how we analyze the diatoms. Well, many researchers have analyzed them also, just extracting them and removing the organic parts and keeping only the biosilica. But again, this will produce some problematics on handling those samples, and that's why we move straight to develop a new combined technique that I will show you in my next slides. But first of all, let me just go quickly through our approach to it. Well, we have a very small collection at INL. Culture collection is just for research purposes. In this case, we don't do any type of upscaling, but it's enough for us. We have several strains for several species growing. And the one we are using more often is the one you see here, which is Cosconodiscus grani, but we have others available. And of course, we have developed a protocol for studying them. Usually, in our protocol, we just start with a little bit of a sample from the collection. We remove the organic components because we want to study the silica first before understanding the whole system, and we remove the parts of that biosilica that we believe might have some type of photonic property. By photonic property, I mean some type of nanostructuration which is suitable to interact with the light very strongly, no? And once we have done this process, we might end up, depending on the species, with something like you see here. This is just one of the parts that form the silica exoskeleton, in this case the griddle band. And if we look even closer, we can see that this membrane has a very well-defined pattern. So this pattern might have many different functionalities during the growth of the life of the diatom. But for us, the most important part is that this very regularly set of porous is actually, they are distributed at a scale which is in the order of the wavelength of the visible light, as you see here, about 300 nanometers. And this is very interesting. I won't get too much into the specifics of the technique of this technique here, but we can easily go using techniques which are already available for other photonic technologies. We can go and check at very small areas in this pattern. This has been applied for plasmonics. I went into the details here, but as you can see, you can go as small as a few nanometers and still being able to analyze the spectrum there. And this is what we have done. This is the system we have. in our lab, as you see, is sort of homemade, although we plan to do this more user-friendly soon. And in general, we have white light coming into a standard microscope, and then we collect that light and we put it into a spectrometer. So as you might expect, what you will get from here is a spectrum of whatever sample you are analyzing there, right? And just very quickly on some test samples. These are nanofabricated samples. These are not yet related with algae, but we just check that we obtain what we expected spectrally and also resolve an angle here. And just to give you an example of a plant instead of a microalgae in this case, here we are analyzing a specific type of chloroplast that are found in some species of begonia. And in this case, these chloroplasts present a specific structure. The thylakoids are arranged in a way that they will produce light interference in a very strong way. And therefore, they will produce this very strong peak in reflectance in the blue. So usually, as you know, you will get a scattering in the infrared, no? In this region here, if you will measure the reflectance of a normal chloroplast, here we get this strong peak in the blue. You can see the details in our paper here, or contact me if you are interested. But the important thing is that we can, with this type of setup, we can go to one of these organelles and measure the reflectance of only one of them. And that's very critical to characterize this reflectance peak. So what we did is we took one single sample of a diatom of interest, and we went and we analyzed these specific properties of the exoskeleton. No? And what we found is where I was mentioning earlier, is that indeed this arrangement will produce a strong filtering effect in this case for in the near infrared. No? So obviously the pigmentation is shows absorption at shorter wavelengths, so higher energies. So we are still in our way to understand what functionalities this filtering effect might have. But as you can imagine, and as I saw earlier, just the fact that we have an algae producing an optical filter by itself is very interesting from a nanotechnology perspective, right? Then if that will affect the yield, for example, of the growth, and that might have implications for biofuel production, for example, is something that we are obviously at early stage, but could be also interesting to investigate. And this is just another confirmation that this effect, that this angular measurement is very specific, but it's very important for us just to understand the system. And also another important feature is that they produce these very strong colors, as you see here, only from the silica, which can be also of interest for some applications like structural color production. And so till now, I've been talking to you about only the biosilica exoskeleton, but as I say, we are interested on the living system. No, we are interested in understanding how the microalgae is growing and using these photonic effects, and therefore we need to analyze as well the photosynthesis activity in the cells. But I showed you before that that obviously we need to take into consideration the biosilica exoskeleton, and therefore we need to go to one single cell and try to analyze photosynthesis activity in situ, while we are also able to measure these optical properties, these other optical properties. So therefore, for analyzing the photosynthesis, we use the standard technique. We use fluorescence analysis for the fluorescence of the chlorophyll in vivo. And specifically, we use PAM, pulse amplitude modulation fluorimetry, that very generally speaking is a measure of the fluorescence when we apply a pulse of a specific wavelength. We measure the fluorescence, and then we, using actinic light, we mimic the light environment over a certain period of time, and then by analyzing those data, those fluorescence data, one can obtain a clear, if you want, a number on the quantum yield of the photosynthesis activity of that particular cell. Now, the challenge here is how to move from the analysis of a large area to the analysis of a single cell. And there are commercial instruments available that many of you might know. Most of them are adapted to work with whole plants, for example, as you can see here. And in a simple image, you obtain all the different parameters through the fluorescence, as you can see in these images here at the bottom. So what we have done is basically integrate that system into the microscope I showed you before, and in a way that we can measure at the same time the photosynthesis activity of each of these cells, and also these optical properties like the reflectance here simultaneously for the same cell. And that way we are now in a position where this is recent advance, but we are in a position where we can start correlating those two parameters. No? So eventually we should be able to tell if actually these nanostructures are indeed affecting photosynthesis or not. And this is just a detail on the extra parts we have added. You can see here this PAM system for the fluorescence. And just some checks with, if you want, non-photonic microalgae. These are microalgae that are being used by our colleagues at INL in the water quality group to check water pollutants, water toxicity, toxics in water, sorry. So we use them, we know they don't have any of these structures, so we know that light will not interact very strongly with the system before reaching the chloroplast. Therefore, they are a very good control sample if you want. And here you can see just an example of the different, so each of these microalgae that you see here in the image, we could go to each pixel and analyze then the activity for each of the pixels. And that is a very powerful information for us, because we can even discern a single chloroplast, as you see there in this example for diatoms. These are actually diatom samples, and you can allocate, as you see here, the chloroplast position in the white field to the photosynthesis activity. And now our next step is to also allocate these photonic properties. No? This is quite a unique, let's say, a unique approach somehow. It looks a bit specific at the moment, but I believe that the information we might be able to gather in the future from this approach could be expanded to other organisms as well, even cyanobacteria that some groups have already started investigating in this direction of the photonic of photosynthesis. No? Just another example of again of chloroplast within the diatoms. And finally, what I was mentioning before, this is the first case where we are able to measure at the same time simultaneously these two aspects. On the one hand side, this is for the high plant I showed you before, this Begonia pavonina. And now you see this chloroplast there, you see the photosynthetic activity through the fluorescence, and simultaneously we are able to measure that reflectance that I told you before was related with the nanostructure, not with the pigmentation of this type of system. So this is, let's say, a proof that we can do it, and now we are really getting into the scientific developments, you know, for this. And just to finish, well, some maybe the most important or the key questions on this field at the moment, and probably the key question are the functions of all these photonic structures. We are finding more and more nanostructures inside the different microalgae, high plants, as I say, cyanobacteria, where one can find these nanostructures. And through understanding their functionalities, we expect to eventually being able to apply this to, for example, biofuel production. No? We need to quantify those photonic effects, so we need new characterization techniques, of course. And finally, we are very interested in bioinspiration approaches. And with this, I of course would like to thank the funding, which is critical to do this, but even more critical, of course, all the people who did the job, people in the group, and all the collaborators involved in these projects. And thank you all for listening, of course. Thanks.
Dr. Martin, so if you allow, we can play the demo clip from our end.
Yeah, if you can play from there, maybe then I can stop sharing. No? Okay. Yeah. Thank you.
Yeah.
Hello, my name is Martín López García, and I'm a researcher at the International Iberian Nanotechnology Lab in Portugal, in Braga. And with this demo, I aim to show you some specific characterization techniques that we use to obtain information on how light is scattered and harvested by some microalgae, in particular diatom microalgae, but I will also show some specific examples for other phototropic organisms. At INL, we are growing several species of diatoms, and also among those 5 to 10 species that we usually culture in our collection, we also have different strains, which will allow us eventually to, of course, implement different type of studies. But just to give you an idea where the samples that I'm going to be showing in the demo came from, we keep them obviously at a small scale. We are interested in fundamental properties. So as you can see here, we keep them in flasks for the different strains. And in this case, I will be showing the example of the Cosconodiscus grani, which is the specimen which is more useful for us because very recently we demonstrated that the photonic properties in this particular species are extremely good, even comparable to some devices, as you saw in my presentation during the workshop. So this is the type of sample we are considering here for the demo. After selecting the strain and the specimens we want to study, of course, the next step is to prepare the samples for inspection under the microscope. And here it's important to know that we won't perform any further preparation than just selecting and taking a few millimeters of the strain that we want, because we want the specimens to be in as close as possible conditions to their natural environment, or at least to the environment where they were grown in the laboratory. Therefore, we will make a very standard preparation for any other study on microalgae that you may be used to, just to inspect under the optical microscope. As you can see here, we just deposit a drop on the glass slide and cover it with cover glass. And after that, we just take it to the microscope for inspection. As you can see, we are using a standard body microscope for our setup, which means that the methodology for inspection of in this case, the diatoms, won't be too different to any other inspection, for example, for checking motility in microalgae. Now, the difference will come on the analysis, but of course also in the type of illumination and the collection that we are using. And those are the most important optical paths that we have modified in the setup, which allow us to collect the optical properties of the living systems, but that comes at the cost of maybe eventually not having such a high quality real image as one would want, for example, for a just for a standard inspection, right? And as you can see here in this example of this Cusconodiscus grani specimen, although the quality of the image might be better, we can still see all the specific features. For example, the chloroplast, which will come very important for our later analysis. But the advantages of controlling the input and the output optical paths is that we can modify, for example, the angle of incidence, which is something we are just doing here, for example, just selecting a very specific angle. And what that will produce is a scattering in this case, in the frustule, in the biosilica, nanostructured biosilica of the diatom. And as you can see here, I pointed it with this arrow. You can see that the diffraction, these colors that are being produced due to diffraction there are very clear. And this is an indication of photonic properties in this microalgae, but it's also a good indication that of the capabilities that the technique allows us. For example, if we can control both collection and also the incidence angle, we could easily inspect what diffractive capabilities these specimens might have, or we could inspect how light is being redirected inside the cell, for example, into the chloroplast. Another very interesting capability is to not only being able to analyze the whole sample, but also to be able to. So then when it comes to actually analyze collecting scatter or transmitted and reflected, in this case by this specimen of diatom, we need obviously to compare in real time the spectrum of the scattered light with some type of reference. So what you can see here is the light collected after being transmitted through in this case, through the diatom, but also through other areas of the substrate, right? So what we want in reality is to select only the areas that actually are interacting with the cell. And for that, we just have a specific iris in our setup that allow us to collect only light from that specific area. And what that means now is that the spectrum you're seeing here on the right is indeed the spectrum of the light scattered only by the unicellular organism. In this case, you're seeing transmission here. And of course, there is a spectral component of the lamp we are using. And that will need eventually, in a further later step of the analysis, to be removed by comparing to, for example, to a diffusive standard. This is a standard procedure, very well known. But the important point is that at this stage, we already have the scattering pattern for transmission of this specific diatom. At this stage, we have seen that we can measure the scattering from individual cells, reflectance and transmission, even controlling the angles of incidence and collection. But now to understand how light is harvested and manipulated by each individual cell, we also need to retrieve photosynthetic activity. And for that, we use a different camera, but as I showed you in the presentation, it's actually connected to the real image camera. And that allows us indeed to see simultaneously the fluorescence of the chlorophyll that, as you know, can be connected with the photosynthetic activity in the cells and will give us a very good indication on how healthy those cells are. And at the same time, we can collect indeed the scattering from each of these cells. So this will be a direct comparison between those two parameters. And I would say this is like a type of measurements that can provide this information. This information might not be useful depending on what you are trying to understand from the sample. But in the case of photosynthetic photonic structures, this is critical to understand. For example, if there is a filtering effect, in this case in the cells, a wavelength selective filtering effect, that will also affect the photosynthesis, and therefore we will see that in our fluorescent measurements. So this is indeed a very powerful approach. Just to finish this demo, I would like very quickly to show you that we can also inspect other organisms of interest with this technique. For example, in this case, the model organism Uglina gracilis, that as you can see, we can inspect again in real time, obtain the scattering pattern from specific areas of the sample in both transmission and reflection. And this data on this organism only strengthens the possibilities of this technique on the study of microalgae in general. And with this, I would like just to thank you for watching this demo. And well, if you have any doubts, just contact me on the email provided on the workshop program.
So now we will take few questions and then end this session for today. So the first question from Dr. Sunil Papi. With your expertise in optics, did you ever try to develop some spectral signature that can be used as identification markers for particular microalga or group of microalgae, or for that matter to access algal contaminants in water bodies?
Yeah, that's very interesting, actually very interesting application. So we are at the moment more focusing, let's say, analyzing at single cell scale, but some of the techniques could be probably upscale to work in the field, for example. And actually, colleagues at INL are working in water monitoring. Is one of the missions in one of our departments at INL, and they are specifically focused on translating some of these techniques that we develop, let's say at lab scale or at microscopy scale, no, into the field by combining the electronics and the optical properties. One of the challenges there is on gathering all this hyperspectral information, no, during in the field, of course, for what specific sensors and specific electronics are needed. I've not specifically involved in that, but there is definitely a large, a huge interest in there for sure. Yeah, and I will be happy to, any optical approach to that, I will be happy to participate in the future for sure.
And the next question by Dr. Zohir. What about microalgae in this botanic activity, and what is the most promising algae for this field? What is the parameter for selecting the suitable and functional algae?
Well, we have found this type of, if we want to call it nanostructuring, no, in more and more organisms lately. I mean, not only microalgae. In fact, I will say diatoms till now have had the advantage or have attracted a lot of attention because the biosilica is, let's say, relatively easy to inspect after extraction from the cell. No? But if I will have to say any, I mean, I found diatom especially interesting because of all the other added values. But if we think more into biofuel production, I think cyanobacteria are very good candidates, although there are not yet examples or so many examples of this photonic behavior in cyanobacteria. But but I think they are very interesting, especially because they are because of the different environments they live in, but also because they the growth is more suitable for this type of studies. So, but this is yet a very I would say it's a very relatively new field, and we are still still investigating the different organisms that could be of interest.
Thank you, Dr. Martin. I would like to extend my deep appreciation to be available at the odd hours and agreeing for delivering to the lecture and the knowledge-filled demo. We appreciate your active exchange of ideas and view on how we could effectively address the optical characterization techniques used in algae and plants. Thank you very much.
Thank you.
Now over to Dr. Ruchi.
So we are now open for discussion. So if anybody has a question, he or she may raise his hand and we will unmute you. So we will address only some of the questions because time is a constraint. So if anybody from the participants having any question, please raise his hand, please. Till then, I can, we can have some questions from our side, which were there in the chat box.
So the question from for Dr. Papi, that why spirulina cultivation farmers are not going for phycocyanin? Who has to promote them?
Hello, am I audible?
Yes, yes.
Actually, phycocyanin, as I conveyed earlier, because it requires some of the investment as well as infrastructure, and that's why the farmer as such is not going for specified products or processing of the biomass, but then they are producing spirulina and then making products from spirulina. But we have a couple of I would say technopreneurs or entrepreneurs who took training from here. In fact, you are right, they are farmers first, and they have started producing some crude products from phycocyanin. They have tried to put this phycocyanin in some kind of oil and trying to, as I'm telling you, some kind of small health related products, maybe for hair and for skin, but those are all products with not very good purified phycocyanin or Phycobili proteins. So the only here is that they are not able to invest so much. And then, as Professor Malik said, that we have not reached that level. So once there is more promotion of such kind of products, then definitely more of the players will come into production of these refined products from microalgae.
Okay. And sir, again for you by Dr. Shovan Mandal, you use different statistical design for optimization. What tools would be best to optimize metabolite in algae based on your experience, and why?
Actually, again, this is depending upon that how many factors or how many products you want to optimize at a time. As the simplest technique which is there statistically, and it is initially applied, is a response surface methodology. But then based on that how many objectives you are going to basically target, depending upon that a particular statistical approach is used, and then which one fits better in the event of the things or the things to be optimized. So based on that, a particular statistical approach is taken up. So as we did for when we were only targeting Phycobili proteins, we went for this genetic algorithm and fuzzy logic methodology. But when we were targeting more than one thing, then we went for multi-objective genetic algorithm. So all these things will vary from approach to how you are approaching a particular kind of optimization. But definitely we have seen that more than one approach is better than taking only a single approach.
So Dr. Shravan has raised his hand and he wants to ask one more question, so we can unmute him.
Hello, can you hear me?
Yes,
Yeah.
yes.
Yes.
Yeah, so I have a query to Dr. Pavan Jatur. So, so you have a fantastic presentation, and I have a query. Like we talked about, like omics approach, and ICGEB have a great actually history for your transgenic approach, and I would say like ICGEB leading this part, like in India, like in the microalgae. But you know, like there is a society challenge that if you grow algae in the outside, the field challenge like from the society, and here like in US, like in environmental agency, they are giving the approval for growing algae in the outside. But India, there is no such kinds of body. So like how this ICGB is thinking, like they can take something like lead to go to ministry and take like making some kinds of agency in India, like to grow genetically modified stuff in outside.
Yes, that is very interesting because as like GM crops or GM algae does not have any till now don't have any particular this thing. So only country I think until now who has approved GM algae for outdoor scale up kind of industrial is Australia. So these are the technical sets. So basically, see, it depends on what you want to grow. So for example, I want to go for biofuel, then definitely open pond systems are the one which is more promising because you have to grow in larger areas. But when you are going for high value products, so you can go for closed reactors because the cost economics can work out on that way. So based on GM until now. So as I said, GM algae itself as a direct product cannot be even taken up approach. But when you extract the product from GM algae that does not have any of those constraints, can be directly taken. The product can be taken. So still, this time I think Department of Biotechnology included some guidelines how to use GM algae. But at an industrial scale, I don't think so. We don't have any open pond-based systems to grow the GM algae.
Okay, thank you. And yeah, so like I just like include one thing like so DBT can like provide like the approval for growing like the smaller scale, like just for testing, like because there is an ecological challenge like you can ask.
Yes, a few labs have the facilities which are funded by DBT or something. So you can get information like that who is working on the like scale. Like for example, there are some bioenergy centers, and also I think IIT Kharagpur also have facilities for scale up little bit bigger reactors. So IIT Roorkee has. So there are some places where you can definitely collaborate with them and go ahead. But I think DBT as such have some grants or projects where you can apply for that. But it depends on where what exactly you want. So you have a 5 liter scale also you can do in a lab which can be feasible to scale up further. So it depends on what is your objective and what you want to look for.
We have another question to Dr. Papi from Dr. Manish Puri. Any live spirulina product available in Indian market?
Could you repeat, please?
Yeah. Any live spirulina product available in Indian market?
Yes, live spirulina, I don't get it. Live spirulina means?
So if we can unmute Dr. Bunish, he can ask him.
Yeah, I'm referring to fresh spirulina. So that is being grown and then directly consumed. So that is well live spirulina. It is quite popular in Europe these days, and in US as well. So that is what I was wondering, that is there any product, since I heard you talk on spirulina, that's why I was interested to know.
Normally live spirulina product may not be available, but definitely the dried biomass is as good as live spirulina. There is no quality differences as such. If it is properly, I would say, dried, there is because live picture will be difficult to store otherwise also. Of course, we have done it. I have visited a couple of production houses, and I have gone there and enjoyed live spirulina drinks and other things. But of course, there can be some ready to serve drinks and other kind of formulations which are available. But as such, biomass, fresh biomass, I don't think there is any advantage of that.
I think the studies have shown that live spirulina has more bioavailability than the powder. So the studies have appeared, and that's why the emphasis at the moment is on live spirulina. So, yes.
About the shelf
It is
life,
based on the bioavailability. Yeah.
shelf life and other things. You cannot keep it for a longer time. Hello.
Hello. Yeah. Can you hear me?
Yes, I can hear you.
Yeah.
Yes, because no, there will be problems with the shelf life and other things because spirulina, cannot be kept for a longer time.
That's right.
And
it is going to be like a ready-to-serve beverage, which is like a fresh or a cold pressed juice. Yeah.
Yes. Those things can be explored. Again, actually, those things are what we have seen. There was some work carried out by one of our collaborators in IIT Kharagpur. They prepared a ready-to-serve drink, but then its shelf life was only 2 months or 2.5 months. So such kind of difficulties are there. That's why more of the products are coming from dried powders.
I may add,
thank you.
I would like to add a little to this regarding this live spirulina used in the food item, that in our parent lab in Banaras Hindu University, in Professor H.D. Kumar's lab, I think 20 years before, we are using a small pot to grow the spirulina, and we prepare like palak puri, we prepare puri with spirulina. That you can, we were using in that way. That directly taking the biomass and putting with our wheat flour and making a puri. That is how you are using it directly. Okay.
In fact, we have also prepared pasta and cookies using fresh spirulina as well as dried powder. But our observations have shown that the dried ones, they are much better. For wet ones, you need to have more biomass. The moisture content is also there, which needs to be compensated. So that's why it is better, the preparations are better, right?
What I will do that, Dr. Papi, I will send you one of the publications, and that could be some sort of a interesting a study which can be looked into. Yeah.
Sure. Thank you.
So the next question to Dr. Martin. I am working with marine diatoms and coppod cultures in laboratory, conducting the toxicity bioassay and developing the seawater quality standards for priority chemicals. control of the ciliates growth in mass culture of microalgae and harvesting the biomass is challenging task. Beside the microalgae are potential for converting the wastewater into green energy. So please throw some light into it.
Yeah, thanks for the question. So yeah, actually, upscaling is one of the critical. In case we want to use these diatoms and other similar species for biofuel or other industrial applications, upscaling is critical. And at the moment, although there has been some demonstrations of bubble bioreactor production of diatoms, for example, is not yet there as for other organisms. So I would say there is some development to do there in the upscaling. In fact, we are quite interested on that, on developing some standards for that, let's say. But at the moment, most of the research has been done in the lab, and that's probably the main gap towards specific applications to solve that upscaling process. So yeah, I guess I don't have the specific solution, let's say, but yeah, that's one of the bottlenecks for sure.
So another question by Rachna. Does the extracellular environmental affects the spectral property of microalgae?
Well, yeah, actually, well, it does, but in terms of if we focus only on the diatom, the way it does, for example, in the uptaking, we know diatoms uptake elements, from the environment into the frustil, into the biosilica. That actually can be used to, for example, check pollutants in water, and indeed it should affect the spectral properties. We don't know yet how. Actually, that's something we would like to investigate in the future. But it should affect those properties according to the theoretical studies, So we are at the beginning of trying to understand if the quantities that are uptaken by the cells will affect those spectral properties. Then in other species, obviously any change in the environment will affect not only the optical properties of the microalgae itself, but also the fact we investigate it, because if we change the environment, we change the refractive index around it in terms of optics, and therefore we need to adapt to that also the way we study it. So there are two sides to it, but definitely the spectrum will change, and we should be able to record it. Yeah.
So the last question to Dr. Papi by Dr. Ravi Kumar. Spirulina cost is ₹700 per kg, while phycocyanin is ₹1000 per mg. If so, why in India technology is unexplored and not reaching the common spirulina pond cultivators?
There are similar kind of questions, so thanks anyway. In fact, the phycocyanin cost will vary. I don't agree that it will be 1000 rupees per gram, or as is mentioned here, but definitely, as we are telling that there needs to be a kind of infrastructure creation, which is the main aspect, and people are not much aware about the applications of these pigments. And most of the spirulina producers are, I would say, small producers, or the ones which are selling the biomass or processing the biomass. And to market phycocyanin is something based on the quality. So that's why there is a lack of producers or so many players in phycocyanin production or processing of spirulina biomass. And rather, I would say, because spirulina is more sold for its other properties of protein and other, I would say, nutritional materials. And that's why if you have seen in my lecture, we are promoting the diazotrophic cyanobacteria, which are very easy to cultivate, very economical to cultivate, and then easy to modulate. So that's why they can be the alternative sources of these biopigments, and they are more robust organisms. Because spirulina production is a bit costlier because it requires carbonate input, and that's why, and then it is more known for its protein and other nutritional aspects. So in fact, the production of these biopigments will be, I would say, see the more importance, as well as more players in the field, maybe in the near future. Let us wait.
Yeah, so as we have very active participants, so we are getting and receiving lots of questions. So we'll be forwarding those questions to our speakers. And with this, I'll say like all the information shared by our guest speakers during last few hours was very relevant, and we have generated a wealth of information and experiences in today's session. We would like to sincerely thank all the esteemed speakers for significant contribution to this workshop, and also we would like to thank you, our participants, for their active participation. Now we would like to end this session with a group photo. Our IT team will be giving you the rights, and you can turn on the camera so that we can have a group photo for this event. I request all of you to turn on the cameras, please. All the participants and panelists and attendees, all of us. So we'll take one more minute, and then we can have a shoot of the group photo. Thank you so much, and we'll see all of us again tomorrow at 10:30 a.m. right here. Yeah, thank you so much, everyone.
Yep.